It's up to 190 hours. So it's really, really nice to not be on Zoom right now. For those of you that are on Zoom, I can see a couple people. Hey, I'm glad I'm not sitting in my kitchen and talking to my dog. It's a really weird thing being on the speaker side of it because you can't see anybody. And I did a program for the EPA that there was over 1,000 people, and I could see nobody. So it's kind of a weird thing. So I'm glad we're kind of moving back to this. Hopefully it sticks. And I was in Ocean County yesterday talking to master gardeners, and so this I'm going to fill up the time today. And I'm trying not to go past noon, but yesterday I was a little bit slow in the first talk.
You're muted and we can't hear you.
I only hear people who are not muted, but not the speaker.
Mary, can't hear any speaker anymore.
Hey, Rich, we can't hear you.
So than any other person in the world. More than 20 hours. Yeah, nuts, right? And then arborists and lawn landscape folks, and we work for residential clients or homeowners as well. Maybe about 80%. And
He needs to get closer to the microphone.
Kids always tell me not to yell. Can you hear me now? Can you hear me now? Yeah, my kid, I butt dialed her on the way in. She's dead.
We can't hear.
Like butt dial. And she texted me as I was walking in the door, especially after she's dead. I thought it comes right.
Ave, Ave Maria.
The hell? It's so funny. They're all screwed up. Nobody can hear them.
Thank you. I thought it was me. Nope, I can't hear anything either. I just heard what sounded like a religious service.
Yes, that's what I heard.
Running off of Mary Cummings, and she is muted. That's where it's coming. That's where he's supposed to be feeding from.
Pests and pathogens of consequence. When we got stuff coming into the country like emerald ash borer or spotted lanternfly, we're part of the frontline survey people trying to stop that stuff. It's interesting because when we do it right, nobody knows.
Thank you.
Right, and when we screw up, everybody knows. Yeah, so it's a kind of a neat program, and there's a bunch of other training things. This Sentinel Plant Network, for instance, is where I use this talk a lot, and that's where we go around the country training public display gardens to educate their clientele. We realized that through the public display gardens, we could get the message out about some of these problems we're trying to suppress. You know, thousands and thousands of people come to the New York Botanic Garden or Longwood Gardens or something, and so we work with them and we help them with educational materials to pass out to the public, and so that's kind of cool because I can fly around the country going to weird display gardens, and you know. I've been to a lot of places I never would have went to, like Green Bay. Who knew they had a lot of reading, you know? So anyway, they help fund the program, and this is a weird one, man. So I'll get used to it. So with that, like, let me just kind of, come on. Now we can't move. Here we go. All right. So what's plant pathology? I'm here to talk about plant pathology. I'm a professional plant pathologist, and pathologists study diseases and all the manifestations of diseases, process development, the consequences, functional manifestations of a disease. You know, what happens to the plants. There's a lot of different kinds of folks working as pathologists. You know, we have people who work as plant breeders. We have people who work on individual pathogens. I mean, there's folks that are experts on one single kind of bacteria or virus or a fungal group or something like that. We got people who look at things at the molecular level. You know, they're molecular. There's a lot of kids that are trained, you know, in sequencing the genome of a pathogen that have no idea how to look at a plant outside and go say what's wrong with it, right? So there's a lot of stuff going on in pathology. My role as a pathologist is a field guy. I'm one of the last folks that was trained in a traditional way. You know, all the kids after me learn from a molecular biology standpoint, and so I'm a field pathologist, and my job is to look at stuff and recognize whether there's a deviation from normal. You know, what's the departure from normal, and that's what you all are going to wind up doing and helping on the helpline. You know, like what's abnormal about this plant, and then can we assign a cause? You know, what happens, and so that's what I do every day, and so. And it's what you're going to be doing too. I mean, I think everyone's a pathologist if they're a gardener, because I think we garden so we can kill stuff. And so you know, that's the fun part. You know? And so, you know, when it's not normal, it's way more. You know, seriously, I went to Longwood Garden with all my colleagues from the Northeast, and all we did was run around looking for dead stuff. You know, it's like nobody cared about anything. Let's brown. Let's go look. So it's that's the more fun. And so we're specialists in this, and so hopefully I could share some of this with you today. And so you know, history of plant pathology, you got to kind of start getting. The first person to write stuff down about plants was a guy named Theophrastus, you know, and he's from Greece, you know, 300 BC, which meant there was a god for everything, and there was a god for weather, and the weather was controlled by the gods, and diseases were brought by the weather, and so we had the wrath of God, right? And so, you know, whenever I talk about that, I always think about Charlton Heston, you know, and we're about that time of year where that stuff's coming on the TV, and so you know, that's all well and good, you know, and I think about that, you know, a little bit, because, you know, what are the people, you know, hundreds of years from now going to think about our ideas about things, you know, and so we're clearly wrong about a lot of stuff. I think we have better tools at this point. Maybe we're closer to the truth, but you know, again, it's interesting how things change. So this sort of led to this idea of spontaneous generation, which is still weather-based. Like, diseases develop spontaneously in their hosts, you know, and even after we started to recognize that there were microbes, in the 1700s, we had microscopes and stuff. We thought that they were a result of the problem, not the cause. So we had it backwards. And so, it took until the 1840s when disease in Northern Europe caused by a pathogen named Phytophthora infestans killed all the potatoes, right? And so all through Northern Europe, they called it the Hungry 40s. We had this failure of the potato crops, and in 1845 and 1846, the failure of the potatoes in Ireland caused a huge problem, the Irish potato famine. Millions of people immigrated to the United States or died. That's your choice when you have no food: move or hope for the best. And so this idea of the Irish potato famine actually started the profession of plant pathology. And so there's a guy named Anton de Berry, and father of pathology. We've got all these fathers coming at us. And where's the mother of plant pathology? And so in controlled experiments proved that a fungus causes late blighted potato in 1861. So it took him 15 years to figure out the cause. And so he was the guy that changed the paradigm from spontaneous generation to the germ theory of disease, right? His contemporaries like Louis Pasteur, got the credit for it, but de Berry really paved the way. And that's how it goes in science. Usually somebody breaks the traditional ideas and changes the paradigm, and then a whole bunch of other scientists kind of fill in the gaps, and so de Berry was right there on the cutting edge, and he named the fungus Phytophthora infestans, which is Phytophthora is plant destroyer, right, in Latin. And it's interesting to me because Phytophthora now is not considered a fungus anymore. It's considered an algae. About 15 years ago, they moved it out of the kingdom of fungi into the kingdom of brown algae. And Phytophthora, it's interesting because true fungi are more closely related genetically to us than Phytophthora is to the true fungi, right? So we had it wrong for a long time, for 100 and some years. But that's beside the point. De Berry started the science, and the thing that de Berry did is what we call Koch's postulates now. And Koch's postulates, Koch was a partner with Pasteur, and they did this originally with anthrax and sheep. And so what happens is, you have a disease plant. I'm sorry that oak tree looks like a mouse, but you have a disease plant, and you describe the symptoms, the deviation from normal, right? And then you separate the pathogen to a pure culture and describe it. And once you get this pathogen, then you put it into a healthy plant and kill it and compare the two, and then take the pathogen out and compare the two. So essentially, you're cycling it through a healthy plant, causing the disease. And that's what de Berry did, and that's what we do. Even today, I just had a paper come out, the first report of powdery mildew on industrial hemp in Jersey. And so what we did was we got plants from the field that had powdery mildew in the hemp trials, and we got healthy plants, and we just rubbed the leaves together and caused the disease, right? And so that was my part of it, then what they did was they got the fungus, and they, this part now is sequencing the genome, and that's how they identify the pathogen. And so again, this is something that happens all the time. And we had to do that just to prove that it was in the state. We have these lists of pathogens that are here and that sort of stuff. But again, this is the fundamental concept on diagnostics, right? This Koch's postulate. Now, fortunately for us, usually we just have to look at this and then look at this and then look on a list. We don't have to go kill stuff, and because people have been doing this since 1860, and so there's lists of pathogens and that sort of stuff. But so again, I just want you to kind of have a feel for what it means to do a diagnosis and get like 100% proof. Now, back to the Irish potato famine, just to kind of sum it up, it gave birth to the science of plant pathology. changed the course of human history. I think this is an interesting idea because the great, movement of humans from Northern Europe to the United States, that human capital, that movement really had a profound impact on what happens here. Like how many of y'all claim Irish descent? Yeah, yeah, right. And yeah, I've got some of it in me, and so it was a huge thing. It introduced like politics. Like, in Ireland, they were still blowing up each other until the Bill Clinton administration, right? Wasn't it that time or something when they kind of figured out to stop bombing each other? And so, they're pissed off like 100 years because the potatoes died, from a fungus. And you kind of think about that, like the impact of a fungus on humanity is huge, right? And so politics aside, you have these two ideas too. Genetic uniformity, that's a problem, right? Everybody knows how to grow a potato, right? Take one big potato and cut it into little pieces, and then you got a bunch of potato plants, right? So if you start with one tuber and then you got 15 plants, they're all genetically the same, right? And so if one can get diseased, all of them can, right? And so that's a huge problem. And particularly, we see like in 1973, it was southern corn leaf blight in field corn, and all the plants had been bred to have this cytoplasm, this gene to make the male flower sterile. It helped in harvest, right? And so all of the corn varieties had this gene in it, and it made the plant susceptible to this disease. Killed like 90% of the corn that year. Yeah, I was little, and so I don't remember how much cornflakes cost, or not even cornflakes. It was probably baked that was more expensive at that point. And so that's an issue that we have to think about. The other issue goes back to the National Plant Diagnostic Network. Problems when new crops are distributed throughout the world. The center of origin for potatoes is South America. That's where those plants evolved from. So you take it to Europe, and in Europe, suddenly it meets this organism, Phytophthora, that's a novel pathogen. It's never seen it before. There's no recognition on a genetic level, and so it's naive, and that pathogen, caused an epidemic. And you ever heard this stuff before? Recently on the news, and so and so that's an issue, and so we're moving crops and we're moving pathogens. Emerald ash borer, spotted lanternfly, chestnut blight, Dutch elm disease, Japanese beetles, all of the significant disease and insect problems in our country are almost all imported. None of them are native pathogens. The native pathogens cause diseases, but we don't get epidemics, right? And because the plants and the pathogens co-evolve together, and they have defense mechanisms, and it keeps it in stasis. Sometimes the fungus wins, sometimes the plant wins, and that's the way nature intended, I guess. But in the modern economy, in the global economy, moving stuff around, things like Queen Anne's lace. That's not native to this area. It's been in the United States for 300 years. And there's some landscape architects say we should give it like native plant status because it's been here so long. But that's an introduced species, and so we have to keep vigilant about that sort of stuff because we got a new one in beech now, a nematode causes beech leaf disease. We got diseases killing all the yolks, killing all the ashes. What's going to be left? And so I think it's something to keep in mind. So at any rate, all of this stuff started to become apparent in the Irish potato famine. So anyway, so what's a plant disease? I think that's the next thing we have to talk about. And so any disturbance of a plant that interferes with its structure, function, or economic value, or any condition of a plant that's contrary to grower expectations, right? So this doesn't say much, except that anything that goes wrong with a plant can be called a plant disease, right? And so, regardless of cause, there's a lot of causes of plant problems. And so I think it's important to think of this in terms of a relative idea. Like, what is your expectation for your plant material, right? A soybean farmer is interested in yield. how many soybeans can I get off an acre, right? They don't care what they look like. In fact, they're happy if the leaves fall off late in the season because you can harvest them easier, and that sort of thing. So that's one thing. Like I work with golf courses. They're all about a game. It's playability. Does the ball roll true on the green, right? And then you think about, Monday I talked to municipal like sports field managers, and they're all about safety. They just want to keep cover on the ground so the kids don't get hurt when they fall down, right? And then you think about like a home lawn. It's aesthetic value. What's it look like? That's all. And how many flowers are we getting from our zinnias? And that sort of thing. So everybody has a different idea of what plant health is and what their expectations for plants are. And so I think one of the hardest things to manage is expectations for plants. I find that folks have unrealistic expectations all the time for some of the stuff they grow, right? And so how do we manage that? I think is one of the things that you want to think about. And the other thing is everybody's got a different idea about what is plant health and what caused the problem, right? So I get this, I bought this arborvitae at Home Depot, and why don't it look like the picture? Or I planted 10 of them and 5 of them died, and then I say, well, I think you did good. And then they hang up on me or something. So nobody wants to hear it, And so again, just think in that terms. Human cost, I guess, is the way to describe this slide. Now, if you think about this in terms of like Anton de Baer, right? A condition of abnormal physiology in a susceptible host plant that's the result of the plant's association with the disease-causing agent with a set of favorable environmental conditions. So it's a more complex relationship, like susceptible host plant, causal agent. 2 living organisms have to be associated with each other within a set of favorable environmental conditions, right? So there's 3 things that are necessary for disease to occur, and we illustrate this with what we call the disease triangle, right? So this is the fundamental concept for plant pathology, right? So this is something I think about like all the time. Every time I walk outside, I'm at work, and so it's a curse. And so because I'm seriously, I'm like, what's the temperature? Is it wet? Look, the grass is starting to grow. I just noticed that you all have all the pears are popped now coming down the highway. We're just on the edge of it up there. So it's a little bit warmer. What samples am I going to get? That it was 24 degrees at my house this morning when I walked the dog, right? And so what is that going to do to the magnolias that popped last week? And so I'm thinking about this stuff all the time, and so should you. And so I'll share my curse with you. And so host, disease agent, environmental conditions. So because this is important, that's what the rest of this talk is about, or the rest of this part of the talk is about, the disease triangle. And so I'm going to start out with this one, host plant condition, right? This is the longest slide in the whole thing. I'm going to ramble on this one. And so, so healthy plants resist disease. They tolerate disease. And a healthy plant generally has the genetic capability to fight back against a native organism, a microbe, right? And so in order for a plant to be diseased, we have to break down that resistance, and I like to call it immune response. That's improper, and because I was doing this talk and I said immune in a program in Connecticut, and this guy from the Connecticut Agriculture Experiment Station took me aside afterwards and was like, you can't call it immune. And he's right, and then I realized that it was his wife that did all the original research on the plant responses on a genetic level, and I thought, well, at least he didn't slap me, like. No, I wasn't making fun of his wife. I just people understand immunity, immune response, not the systemic acquired resistance pathway, or and so it's like salicylic acid turns on all these switches, and then the plants fight back. And like trees will make like volatile compounds to gas spores that are trying to come in. or they'll kill themselves around an infection site, you know, and then you get leaf spots, and that's a problem because you're going, oh, there's leaf spots, but it's not a problem for a plant. It stopped the infection, you know, and so there's a lot of different mechanisms that happen in plants, and so how do you break down these mechanisms, or how do these mechanisms break down so plants can get invaded by these organisms, and so when the organisms win? So I kind of think of it like this: resistance and susceptibility are sorry, I just got a text from myself. That's really weird. And so that has to be spam. And so so you have resistance on one side, susceptibility on the other, and it's a continuum. It's not absolute. Down here, immunity is absolute. That's something different. I'll get to that. So resistance and susceptibility, and so there's a genetic set point in there somewhere. And like we have plant breeders trying to breed resistant varieties, right? So we get a better set point over here toward resistance on this side, susceptibility on this side. Hopefully I didn't change which side is which, but you have a genetic set point, and that's a good thing. But the environmental conditions can kind of move it off the set point, right? And weaken the ability of the plant to tolerate or resist. And so I think the best example, I always use a turfgrass example for this. So I could talk about golf course putting greens a little bit. And I just put in my grades for the professional golf turf management school. And my students are stupid. And so I think it's the Zoom format. All the grades are at least one letter grade lower, like my bell curve. I've been doing it so long. I always get a perfect bell curve for my grading system, and it's moved like down, and it has to be Zoom. So maybe it's not that. It's just the format. But anyway, so we breed turf grasses at Rutgers, right? Rutgers is the premier turf grass breeding institution in the entire world. We have more than 350 varieties on the market and something like 30,000 germplasm selections, wild type grasses that provide genetics for the breeding program. And so we breed for golf courses. And one of the main things that we breed for is resistance for dollar spot. And dollar spot is a disease of turfgrass. I think it's the most important turfgrass diseases. On golf course putting greens, they put out about $60 million worth of fungicides every year for dollar spot control. So on a high-end golf course, you know, something like Pine Valley or something, a high-end, what do you got down here? Marriott Seaview, you know, something over there, Atlantic City Country Club, they're pretty high end. They're going to make 12 to 14 fungicide applications on the putting greens every year for dollar spot control, right? At about $3,000 to $5,000 a crack, right? Yeah. So thinking about that, right? And that's in just a normal preventive program, not if they're having outbreaks and stuff. And so then the kitchen sink goes out, and like. So if we got a grass that doesn't get dollar spot or tolerates it well, and we can cut out some of those treatments, instead of going every week or every 2 weeks, go every 21 days or 28 days, you cut out a few treatments, you're saving a significant amount of money. And when I talk about that to golf guys, if I can, you know, they don't want to hear the environmental thing. They want to hear how much money can I save. That's the angle I go with them. And so landscape guys too. And so what's it going to cost? You know, and is it worth it not to spray? Is my plant going to die? Anyway, so we got these grasses that have dollar spot resistance. On our turfgrass research farm, we treat them very well, right? They're mowed at 0.156. That's 5/32 of an inch. And that's pretty tall for a putting green. One of my former students at Arcola Country Club up near Paramus, he mows twice a day at 0.08, right? Half the size of what we mowed on the turfgrass farm. And if you reduce that leaf surface area, you're reducing I think of leaves as solar panels. So the smaller the solar panel, the less energy you can make, right? And the less energy you make, the less storage you have in carbohydrates in the roots. So the roots are shorter, right? So the smaller the grass is, the more resources it needs. there's research from Nebraska that says the shorter that you mow grass, the faster it grows. So the short grass grows faster than the tall grass and needs more resources, but it's really stupid to put more resources on cutting green, right? You don't want to fertilize because that's going to give you top growth and slow the ball down. And you don't want to water because you want it hard so the ball rolls like a pool table, right? So my interns from the turf school, what they do all summer is they stand by greens with a hose. And like they see the hot spots forming, they go hand water the hot spots all day long, up to sundown, 100 days in a row, you know, for an internship. And you have to do it right. You got to hold the hose right, because if you hold the hose wrong, you'll kill the grass. It's a hard thing. There's a skill set to watering, you know, grass that's about to die, you know. And so I had a friend, a former student who became a friend. She's a golf course superintendent on Long Island now. And she was so strong, she could carry 100-foot hose and run between golf greens. And like, I couldn't believe it. And she always said, try to throw me down on the ground. Like, I'm like, no, I'm not touching it. But anyway, so you think about that, right? You got a plant that's close to dying because it doesn't have capacity to make its own food, right? And you won't fertilize it, even though we eat it badly. And then you won't water it, all for the game of golf. And so this resistant grass suddenly has dollar spot, right? It's not dead to the dirt. There's bent grasses that don't have the genetic set point that will be completely dead. There's one called Crenshaw. They can grow it in Texas because it's bred for heat, not dollar spot. So it gets macerated by dollar spot here. But so you still have to spray. Maybe you spray a little bit less, but you still have to spray. So if you guys are like landscape guys, I would be telling you, you'll never have dollar spot in your landscape if you mow at the proper height, you know, if you fertilize according to soil tests and the turf grass needs, and if you water with precision, if you give it the food and water that it needs and allow it to grow so that it can make its own carbohydrate, it's always going to fight off dollar spot. You'll never have to spray fungicide. Right. And so that's the message that we send to most everybody about turf grass. If you grow it right, you don't have to worry about fungicides. Never spray fungicide on your lawn. Right. You know, the only reason we have to do it on a putting green is because they're growing grass. Their expectation is for a game. It's Disneyland for grass. You know, it's fake. And so those of you that are appalled by the amount of fungicides they might use, recognize that a golf course is about 25 acres and the greens are only about 2.5 acres. So it's not that much turf area that's getting sprayed like that, just the playing surfaces. So anyway, so you get the idea, right? There's a little bit of fluidity and resistance. It's not absolute. Immunity is absolute. If the plant don't get it, it don't get it. Not all diseases get on all plants. Not all plants get all diseases. So that's an important concept for us in the second part of this talk, because one of the things that we do is we use plant associations. What plant is it, and what do we get? You know, what do we have to pick from? And so that helps narrow down possibilities. All right, so we have two types of causal agents. We have living organisms that cause infectious disease. You know, the pathogen grows, multiplies, spreads. We have non-infectious problems, environmental conditions that kill. You know, heat, drought, all that stuff. I like to call them physiogens compared to pathogens. Largely because I'm just remembering my old professors, Dr. Dave Lewis and Dr. Lewis. When I first started, I'd go up to him and ask for help. He was a vegetable guy, and I'm like, you know, can you help me? What's wrong? And he'd go, that's an unknown physiogenic disorder. And I'd be like, okay, what do you mean by that? He's like, I don't have any idea what's wrong with that plant. You know? So if you don't know, make something up, you know, and I can write a pretty good letter saying I have no idea what's wrong with this plant, you know, so physiogen, pathogen. Look, 90% are injuries. I don't like to call, some of the textbooks call them abiotic diseases. I'd rather call them injury because I think disease means whole triangle, right? And the abiotic causes are the environmental part, right? Now they will play a role. These environmental causes play a role in disease outcomes, but they can kill plants outright too, you know? So we got to recognize that. Look, 90%, 10%. I got that number from a textbook that George Agrios wrote, Introduction to Plant Pathology. And that's the one that all of my generation used in you know, undergrads and stuff. And Dr. Agrios, I remember one time I was sitting next to this old dude at a meeting, you know, an old crusty guy talking to him. You know, now I'm the old crusty guy, but you know, and I was like, I had no idea who it was. And we had a long conversation, and then somebody was like, oh, I saw you talking to Agrios for like half an hour. And I was like, oh my God, that was Agrios. And then I felt really stupid, you know. I talked to the guy who wrote the book. And so, but Agrios, you know, Agrios is wrong, and he's right too about these numbers, because if you look at our data lab, my data, this 90% number is 40%, right? And so if I talk to my colleagues in other states, my friend Lee Butler is at North Carolina State, and he's always like, it's always between 35 and 45%. And if we go back, I have data going back to 1991, and it's always between 35 and 45%. Same as Lee, University of Delaware, you know, Oklahoma State, Michigan, everybody's all in that range. And I think the reason is because we are dealing with pretty sophisticated clientele. You know, Lee is a golf guy, so he's getting all golf guys down there. And I got golf guys walking in going, I got dollar spot. And I go, yeah, you got dollar spot. And they just want a letter that says, you know, spray it 14 times and spend 50 grand, you know? And so that justifies their decision making. Or I got a guy in Princeton. This guy, he's been a tree guy since 1967, right? And he comes in my lab and he hands me plants and tells me what it is. And he's right like 90% of the time. He's right all the time. And like, say, why are you coming in here? You know, what's it, because of the air conditioning? Because you're like 80, you know? And he's like, no, he's like, you know, my clients don't believe me. After all these years, they don't believe me. And your letter gives me credibility. And then he's like, I just charge them your fee anyway. So you know, so I think that's what skews me down. And so when we start taking out those professional clients that have an idea of what's going on, and we work our way down to homeowners, 90-10 shows up in a big way, right? So I think the lesson from this slide is when you're deciding something for a residential client, you know, if you're a landscape manager or something coming onto a property for the first time, I'd be looking at the abiotic stresses first: heat and drought, planting problems, poor plant selection, stuff like that first, and then go toward the diseases, right? So you can rule that out, and that we'll talk about a little bit more too. Just to kind of beat this up, talking about these causes. I love this picture. This is on Rutgers campus, on Cook College campus. And it's like, whose idea was to plant trees in parking lots? You know, because it irritates me to get, I hate parking lots where you can't park in half the spots, right? And you know, there has to be a school of parking lot design somewhere that we can figure this stuff out, right? And so thinking about this, like, how do you make a parking lot? You know, there's got to be some like leveling and some scraping and some steamrolling and some impermeable layers. And what's that do to the growing conditions? You know, we have a root space issue, we have a compaction issue, we have a drainage, it's too wet, too dry, we have a heat sink problem. You know, we have everything coming out of this car going in that hole. We have de-icing materials. You know, I read an article in the Philadelphia Inquirer about Jenkintown. They were measuring the salt level in the water after snowstorms, and the salt levels were saltier than the ocean, right? And the pH levels of the streams are going up from calcium chloride too. So it's like there's a pollution issue, you know, and then the big pile of snow is on top of the tree, and all the shopping carts are jammed underneath there, and that dude, look at that guy driving a war wagon. You know what happens when he pulls in? You know, I used to drive something like that. If I backed up, I just drive off. You know, I don't care. I hit anything. You know, you know, that's parking in New York City. You know, you know, I can fit in the spot. You know what I'm saying? And so like nobody cares if they hit that tree. And so, you know,
the thing is here with a picture like this, if your expectation for that tree is it to make it till it's like 70-foot height and 100 years old, you're out of your mind, right? You know, you know, and I'm teaching these kids, they're like 18, they're out there smoking in between classes. I'm like, you guys got to quit smoking. And I always got a kid that says, oh, Uncle Jake made it to 90. He was smoking Camels and drank Jack Daniels for lunch every day. You know, and I'm like, yeah, but Uncle Jake's buddies were dead 30 years before him. You know, he's the genetic anomaly. So you're gonna get one tree that survives, you know, but most of them are gonna be dead, right? And so if your expectation is that they're gonna die, right, then that's okay, because commercial landscapes very often are designed to be replaced every 10 years or so. So you plant and you yank it out and you plant again, and that's good. That's good. That creates jobs, right? There's guys have to grow them trees and guys have to plant them and pull them out and put them back, you know, and that's okay. That's a real expectation. Have you ever been to Disney World and seen a dead plant? Right? That's because there's like 10,000 people at night running out and replacing everything, you know? So no, seriously, I had a friend and her job was for the Six Flags group. And she was in charge of like the landscape management stuff. And then basically that's what she said. We have acres of greenhouses and we just replace everything every day, like constantly. And so we didn't care about pesticides. We'll just get a new plant. And so we got trees, you know, and so that's okay. So, but if you want this to live, you've got to pick something that's going to suit that site, right? We call that tree of heaven, you know, and yeah, right. That's going to work there just fine. So you kind of get the idea here where expectations and all that start to play. And so that's the abiotic cause, physical, chemical, and mechanical. We'll talk about them a little bit more later. What about the living organisms? So we have fungi, bacteria, viruses. Viroids are kind of like viruses, you know, if you can believe there's a simpler form of life. Molecules are simpler forms of bacteria, like walking pneumonia is mycoplasma. You know, that's a human disease. It's a molecule, and we have them in plants. We call them phytoplasmas in plants. Protozoa, there's some weird diseases in turf, one called rapid blight. It's a protozoa. Algae, you know, we have phytophthora, we have pythium, we have downy mildews that are considered algae now. We have cyanobacteria that actually causes nutrient deficiencies in turf grass. You know, so that's kind of a weird thing too. You know, insects and mites meet the disease triangle model really well. In my world, they're just pathogens, you know, just another pathogen. And guys will come in here thinking that they're different, you know, but, you know, insects kill plants too, you know, they're just easier to diagnose. About 11% of our samples have insect problems. And so, so that's something to keep in mind. Parasitic plants and then nematodes too. And so I'll start with fungi and kind of elaborate a little bit on this stuff. We've been studying fungi since the beginning. Fungi are everywhere. They are really important organisms in our ecosystems, in our forested areas. Some of them are really beneficial to plants. Others are not so good. You know, they rot dead stuff. And fungi, that's their primary role, is to rot dead stuff. And some of them rot plants that aren't quite dead yet, is kind of the way I look at it, and the susceptible ones. And so they're most important as agents of decay. And because they decay things, we can exploit the byproducts, you know, like beer and pizza. You know, fungi will, you know, break down sugars, and you get carbon dioxide, which leavens bread, and then you get alcohol, right? And so that, you know, and cheese, what's cheese? Just rotted milk, right? You know, the bluer and rottener it is, the better it tastes. You know, that's when I was little, I was gonna say when my daughter was little, when she was 7, 8 years old, I used to try to get the stinkiest, nastiest cheese I could find and see if she would eat it. Like, and you know, it was a game that we always played. And so anyway, I was just trying to train her for the future. And so fungi are good, right? I don't know if you ever watched on Netflix Fantastic Fungi. Yeah, it's kind of interesting. Paul Stamets is kind of a hippy dippy dude that does, he's not a PhD, but he has a organization in Canada that does some interesting actual research, and but he's a good kind of colorful commentator. It's an interesting thing to watch. I watched it with a grain of salt. I'm like, nah, you're talking about something, but you know how it is. And like my dad used to watch sports. He was a coach, and he'd be yelling at like Howard Cosell, because Howard Cosell didn't know what he was talking about. Because yeah, anyway, I tried to get over that, and so but it's hard when you're watching the Fungus show. And so anyway, they can tack wood, leather goods, fabric, petroleum products, foodstuffs. They rot stuff, and there's a fungus on the inside of the Chernobyl nuclear reactor on the start rotting the concrete on the inside. Yeah, that's wild, right? And so how can something be alive in there? And they think it absorbs gamma radiation, like plants absorb, light. And so eventually it's going to rot a hole, and I don't want to be there when it opens up, right? But that shows the versatility of the group. And so they infect animals and plants. I think everybody's, had some experience with fungal infections of some sort, and on your feet or some other body part. And so most common of the plant pathogens. So when you see a fungus, right, what you see is a mycelium, and that look, this is Pythium, so it's really an algae, but it's the best picture I have of the water molds that are algae now were fungi, forever. And I'm going to call them fungi because that's what they were called when I learned them. And, that's another thing. One of my old professors, Phil Haliski, he used to tell me, he's like, that's what it was called when I learned it. That's what I'm going to call it now. And I thought he was crazy. And then 30 years later, I'm saying the same thing, but mycologists still study these organisms, even though we've moved them, right? And so we still can call them a fungus. We can't call them fungi because fungi is the kingdom, and it's not really in the kingdom of fungi. This is confusing, right? And so here's what you see. You see mycelium, which is singular, but that's the mass, right? So mycelia is what we should be calling it, but in practice we call it a mycelium, and the individual strands are called hyphae, right? Which is plural, but it's a single strand, and one is Latin hyphae, or one is Greek hyphae, and one is Latin. So it's all confusing, but what you see here is the mass, and that's the body of the fungus, the vegetative growth, and individual strands are hyphae. These things are female reproductive structures. This organism is what we call homothallic. So it mates with itself. There's a male structure and a female structure, and they mate, and it results in a spore called an oospore. And the oospores survive through the winter or in the absence of a plant. And when they germinate into a structure called a sporangia that makes a zoospore. And zoospores swim, right? And so when the zoospore gets to the plant host, it turns into a cyst, and you get a hyphae. And the hyphae is a germ tube that grows into the plant. And when it grows into the plant, it grows in between the cells. And so what it does in between the cells is soak up nutrients and water that's moving between the cells. And that causes in the plant a dry rot, right? You're going to see a spot that's dry. And that's something that we're going to come back to in the second half when you start to recognize and try to separate the difference between a bacteria and a fungus, on a plant. And so think about that, dry rot. And so it doesn't break the cell walls apart, and just soaks up the nutrients. And fungi generally grow in concentric circles, right? So you're always going to see rounds, right? And so that's another kind of a clue. And so what I'm trying to get at here is getting you thinking about how these organisms interact with one another. And then you can use that idea of pathogenesis, to make a determination. This is a fungus, this is a bacteria, this might be a virus, something like that. So then let's go to bacteria. So bacteria are saprophytes. That means they eat dead stuff. right? Parasites, live stuff, saprophytes, dead stuff. There's switch hitters in between. You know, you almost could call some bacteria switch hitters. They kill their host with chemicals. They don't invade the host and get food. They kill it first with chemical warfare. And we call those chemicals antibiotics when we use them for our benefit, you know? And so these are important organisms. Bacteria are everywhere. I mean, by some accounts, about 10% of your body mass is bacteria, which, you know, me at 165, that's 16 pounds of me is bacteria, right? Think about 16, that's like a bucket full, right? and nasty. And so you're never alone, you know? And so, and like, we have a whole bunch of people trying to figure out what this bacteria is doing for you. You know, it's doing stuff for you. And you hear, you know, if you listen to Gwyneth Paltrow or something, she's going to tell you, buy my product because it's got bacteria in it. It's going to make you feel better, you know, and or something, you won't have gas or something like that, you know, and that's true, but I don't think that we understand it well enough to be able to buy a pill and fix it, you know, but there's a lot of stuff that bacteria do that are beneficial for plants. You know, they help fix nitrogen, they help recycle nutrients, they help plants take up stuff, they help plants. There's bacteria on plants that help plants get ice over top, ice nucleated bacteria that we can actually spray on strawberries that irrigate and freezes them all so they don't die in the cold, you know, stuff like that. And so you know, it's interesting the good things that bacteria do. They also cause a lot of disease, tuberculosis, pneumonia, typhoid. You know, the plague was a bacterial problem. Tuberculosis really would, you know, if we didn't figure out like what antibiotics were in the 40s and 50s, like half of us be dead from tuberculosis already. You know, that was a pretty significant problem. Anybody that wants to go back to the good old days, you know, go ahead, because I'm not catching smallpox or you know, any of that now. And so so these antibiotic compounds, I guess I got to mention actinomycetes, right? Produce antibiotic compounds. So these organisms, they defend themselves with chemicals, right? They communicate with chemicals. In the world of plants and microbes and insects and stuff, it's all about chemicals. And so you know, the first antibiotic was penicillin, right? Flemmer, you know, discovered that in the 30s, I guess it was 30s. And then Selmer Waxman came along in the 50s, who was at Rutgers in the soil science department and discovered streptomycin sulfate. And that was the first product that we could use that actually was effective against tuberculosis. And it's an important thing because, you know, that research group, you know, we got a lot of stuff out of that. Those actinomycetes produce products that we use as insecticides now, products that we use as fungicides now, products that we use in human health. A whole bunch of the antibiotics are based on bacterial toxins, things like ivermectin. You know, you see that in the news that you give to your dog for heartworms, right? It's a nematicide, is a actinomycete based antibiotic, right? Boyd Woodruff was at Rutgers, and he went to Ciba-Geigy, I guess. They won the Nobel Prize for that in 2015. Waxman won the Nobel Prize, you know, for discovering this. And it was a huge thing because it had a huge impact on human health. And so bacteria are good and bacteria are bad, same as fungi, right? They reproduce by fission, right? So this is a bacterium called Xylella fastidiosa, and they divide in half about every 20 minutes. It's xylem limited. It's in trees, and it breaks apart about every 20 minutes into 2 equal clones, and that means one turns into 17 trillion in like 24 hours, right? So you know, there's bacteria everywhere. There's billions of bacteria, and in the soil, you know, you're going to have billions in 1,000 square feet, right? It's really interesting. They live in colonies that protect themselves with like a sugar solution that we call an extracellular polysaccharide ooze. And there's thousands and thousands of individuals, and you know, that's on the head of a pin in these colonies. And so if you look at them in plant material, you know, this is streaming out of a bentgrass leaf. And so these are individual bacterial cells that we could see, you know, it's called a phase contrast microscope that bounces the light in different ways. There's 400 times magnification to see them. And it's cool. But, you know, I had a kid working for me named Lisa Byrne, and her PhD in plant pathology was to look at the impact of pesticides on soil microbes, right? So what Lisa did was she collected soil from all over the world. She had rainforest soils, she had boreal forest soils, she had soils from the desert. She had 19 different sampling sites, and including the Rutgers Turfgrass Research Farm that has been golf course research for like 80 years. You know, so it's getting all kinds of pesticides and fertilizers. And she used a technique, metagenomic analysis. And what that is you can blast samples and detect all forms of life, right? And so one of the things that they started doing with that was like the World Sea Project, where they got ocean water from all over the world. And like in the Sargasso Sea, there was something like 1 million viruses in a gallon of water, right? So what Lisa found out was in a gram of soil that there was more than 600,000 unique living organisms in the soil. 40% of them had never been described before, right? But the most important thing that I thought that Lisa said was that no matter what sample she had, rainforest or golf course putting green, the results were similar. There was no statistical difference in numbers and diversity of species. So it kind of put the rest of the idea that pesticides sterilize or damage soils, right? And so what they found was that they could change different nitrogen sources, and some of the organisms would increase and decrease according to what the food source was, but it was usually about 48 hours and it goes back to stasis, right? So when you put fertilizers and pesticides on soils, it's somebody's lunch, right? You know, and you know, we get a lot of like blowback about pesticides and that sort of thing in agriculture fields and stuff like that. And the reason why that you may have less culturable organisms is because, you know, the number of microbes in the soils is depending on carbon and oxygen and moisture, not what kind of pest toxin is on there. The toxins is just another carbon-based food source. So I think that was an interesting thing. And it goes back to this idea that I don't think we can hurt or we can help. You know, I don't think we can affect it all that much. And that goes back to Gwyneth Paltrow and your stomach. You know, I don't think we can hurt or help that much either, you know, but again, it remains to be seen, you know, because we have these new scientific techniques like metagenomic analysis, you know, we can start to measure stuff that we couldn't 15 years ago. And I think that kind of goes back to the Theophrastus and the Berry. And, you know, as we develop new tools, we learn more stuff, right? And that makes some of the old stuff obsolete. It's an interesting thing. Science is a process, not a set of facts. And so all right. So am I pontificating? Yeah, I think it's fun. You know, I love getting in arguments about it. So, in Monmouth County, we had a master gardener that put like "Stop Monsanto" bumper stickers on everybody's seat, and yeah, so if I was talking about that, I'd be getting yelled at now. So you guys stay calm. All right, viruses. No, viruses. Anyway, entity that contains genetic material within a protein coat that can only reproduce using the metabolic processes of a suitable host cell, right? So viruses aren't really considered to be alive. There is some argument over that, but they're not alive because they don't meet the general rules of living organisms. They don't reproduce, they don't eat, they don't grow, they don't really do anything except boss people around, right? They boss other cells into making copies of them. So they commandeer cells and say, make a copy of me, and that's what the cell does, and then it doesn't do its job, right? I think everyone remembers when we first got our first computer, right? And there was solitaire. right? To try to teach you how, you start playing solitaire at lunch and then it's 3 o'clock and you didn't do anything, right? So there's a downside. Your family's going to be pissed because you didn't come home, right? Or your boss is going to be pissed because you didn't get the work done. And so that's kind of what viruses are doing. Like, they're distracting everything from their job. So enzymes and proteins and cells aren't being divided. And there's a failure in the host, and in plants, that's usually abnormal color and growth. So that these things have a protein coat and they have a nucleic acid core, right? Most of what we have in plants are RNA viruses, single strands. They're too small to see, can't see them, need a electron microscope. There's different shapes, many of them have more than one. We have Gemini viruses in plants that they're two of them. They travel together, they hang together. There's a whole bunch of viruses out there in plants that we have no idea what they're doing. We have no idea. There's lots of viruses that we can detect now with this metagenomics that we don't understand what they're doing. And so I think that's kind of cool too, a little bit scary even. And so, generally what we're looking at here are polyhedral viruses and you capsid and the nucleic acid core, stuff like that, helical or flex rods. There's some viruses that have a lipid coating. COVID is one of them, an envelope virus. We have some of them in plants too. And so the capsid is you always hear the spike protein, but it's sticking out of a lipid coating. And so again, a little bit different structure. This one is tobacco mosaic virus, and that's electron micrograph. And so it's a flex rod, and you can see the protein code pretty easy. This is a pretty famous virus because one, it gets in about 600 different kinds of plants. And so if you're holding tobacco products and then holding susceptible plants, you can transmit the virus to the plants. Like I have cigarettes in my lab that I've had for like 25 years. And we even got cigarettes from a Chinese grad student from China, right? Chinese cigarettes. And so every cigarette we test, when we have kids coming or visitors in the lab, and I want to show like antibody-based test kits, or, we do a little demonstration, who's got a cigarette? If no one does, then we whip out the Chinese cigarettes and they have tobacco mosaic, and they all, so this virus is rigorous enough to survive the tobacco drying and cigarette manufacturing process, right? Not all of them are that good, but you're going to be careful. Like nicotine's a good insecticide, so like you'll take, red man chewing tobacco, macerate, shake it up in a jar and spray it on your plants. You're probably spraying it with tobacco mosaic. So you want to be careful about that. I had a grower, they threw out 50,000 pepper transplants because they were all smoking when they were spotting them out, and they got the disease in there. Like, at Rutgers, if you're smoking, you walk into a greenhouse, you'll be forcibly ejected, they don't want you in there. And so it's a good virus for plants. The other good thing about this is in the 50s at the University of California, a guy named Linus Pauling was using tobacco mosaic and taking pictures of it. Like he was using this X-ray technique to take pictures of it because they were trying to figure out the structure of DNA, right? And so Pauling couldn't figure it out, but a woman in London named Rosalind Franklin was watching Pauling's like protocols and took pictures of cells and got a picture of DNA and showed it to her boss, a guy named James Watson. And James Watson showed it to his buddy Francis Crick, and Watson and Crick, made a of the structure of DNA, the double helix. Interestingly enough, they won the Nobel Prize and didn't give Rosalind any credit. Without her, they couldn't have done it. So she should have got the prize too. James Watson wrote a book called The Double Helix. And I found it in a trash can in New Hope, Pennsylvania. I was walking, and I grabbed it, and I took it home and read it. It's interesting because it talks about science and ego and the race to discover DNA. But it's also, he basically apologized to Rosalind the whole book, like, we left you out, we didn't give you any credit. And you win like $1 million, and in 1950, that's a lot of money. And James Watson is the only person ever to sell his Nobel Prize medal, the only person ever to sell the medal, and he got $4 million just for the medal. right? So, you should have kicked back to Rosalind then, that's the way it goes in science, I guess, and women in the 50s, and, I sort of gets that. And so anyway, I'm a trained golf course superintendent, right? And there's 15,000 of them in the country, and there's about 100 women golf course superintendents, and I've trained probably 40 of them. So it's kind of wild. I had one in the fall who's in active military, and all the dudes were scared of her because they're like Amy knows how to drive a tank, and you're looking at her Zoom, and she's got all these deer heads and all this stuff. And it's like, yeah, Amy can shoot too. And so anyway, so we've come a long way, but not far enough, I guess is what I'm getting at. And so aphids are virus vectors. So viruses need to be moved, right? Tobacco mosaic, we're moving it with our hands, but most viruses need to be moved with an insect. Right, and usually an insect with a piercing, sucking mouth part, and so that's one of the things that we use. You see a funky looking plant and it's got a lot of aphids or thrips or leaf hoppers, then we think virus, right? So it's a little piece of circumstantial evidence. A couple more of these nematodes. Nematodes are really cool. There's 500,000 named species. It's the second biggest group of organisms next to the insects. There's several thousand species that are plant pathogens. There's a bunch that are in animals. Heartworms and dogs is one of the most common. There's a bunch of nematode diseases in Africa. Interestingly enough, the Jimmy Carter Foundation is responsible for solving going back to presidents here, you might hate him as a president, but as a humanitarian afterwards, he's a pretty good dude, right? And there's a nematode in Africa that lives in the water. And so when the Bushmen have these straws, right? And they just like drink out of like the puddles and stuff. And you drink this nematode and it gets in your body, it moves into your eyes and causes blindness and stuff. And so what the Jimmy Carter Foundation did was they gave everybody a straw with a screen in it that catches the nematode. And they reduced the number of people that died from the disease by like 200,000 a year, right? So just by giving out straws. So it's like public health stuff. It's really cool. And that's kind of, in my world, all these diseases, these pathogens that get in humans, it's the same as get in plants. And we kind of think of it like this. It's like the same recipe, different cake, right? Like like when COVID started coming on, I started reading science and like, well, it just sounds like we're talking about soybeans, and so it's an interesting thing. And so several hundred species feed in plants. We do some nematode work with golf courses. We look at blueberries. There's nematodes in blueberries, so I get a couple hundred samples of IPM sample blueberries. We look at nematodes in grapes. Grapevines. There's a lot of grapevines going in the ground in New Jersey, and there's a nematode that transmits a virus. And once you get the grapevines in, you can't do anything about it, right? So they test the soil for nematodes to see if that critter's in there, and if it is, then they fumigate before they plant. And that solves the virus problem. So you may see nematodes here in a helpline situation or something like that. Root knot nematodes in vegetable gardens are sometimes common, particularly if you're growing like old tomato varieties. Everybody likes the heirloom varieties, and some of them don't have resistance. Some of the newer varieties do. I forget which one Sabrina grew in her garden, and it got hammered by root knots. And none of the other tomatoes got them, so they had the resistance gene in them. But they're not that common as a problem in general. They're common for specific issues, and so the ones that get in plants have a stylum, and so that's a piercing, sucking mouth part, and they feed on the roots and they cause the roots to fail, and the plants suffer accordingly. So nematodes. And then the last bit, parasitic plants. Epiphytes are things that just grow on top of plants. You know, you get a lot of Spanish moss, and that's a problem if you're in South Carolina, I guess. Mistletoe is a hemiparasite, which means that it has chlorophyll and that it takes raw nutrient from the plant, but it doesn't take processed carbohydrate. So it doesn't do that much damage, right, to the tree or whatever that has it. True parasites like dwarf mistletoes have no chlorophyll, and they'll kill the woods. Out in the Rocky Mountain areas, there's mistletoes that kill like pine trees, and they're devastating problems. You know, we have dodder around here. It looks like silly string. You know, I'm talking about like it looks like someone took some yellow fishing string and put it all over top of a plant. And you know, I see a lot of that growing in like drainage ditches and waste areas, sometimes in farm fields. But mistletoe is the picture I have. And you know, the bird eats the seed, puts the seed on another tree. The seed germinates, has a structure called a hostorium that goes in between the cells, and then it just starts getting water and nitrogen from the host, and then it grows. You know, something like this. We don't have it here. I don't think you have it here. Below the Mason-Dixon line, right? I'm from Dover, Delaware, so we're almost close in latitude, you know. And here's another personal story. My first job in the green industry, I worked for a Christmas tree grower when I was 14, and I got paid $1.65 an hour. That was minimum wage, you know. And so I'd take my shotgun out in the woods and shoot trees and knock the mistletoe out, and then bag it up with little red ribbons. And so when I was like, I'd cut down your Christmas tree and drag it to your car, I'd be side hustling you some mistletoe on the way to the car, you know, $1 a bag, you know. And so, yeah, big fan of mistletoe. It was, you know, $1.65 an hour. You know, I told that to a bunch of Christmas tree growers and started getting job offers. So anyway, that we do a lot. I spend a lot of time with the New Jersey Christmas Tree Growers Association. They're a fun group. Anyway, so they always have a food truck that goes to the Philadelphia Eagles games at their meetings, right? So they got this dude making cheesesteaks and stuff, and it's like they spare no expense. So anytime they invite me, I'm like, yeah, I'll come. So, yeah, they're fun. And, so anyway, all right. So what about causal agents, right? In order for the disease to occur, the pathogen must be present, right? And so that means you have to have it. And that's a dumb thing to say, but you know, in the era of Google, you know, people come up with stuff that they can't possibly have. They come in my lab with diseases that, you know, that are like, you know, in Brazil and have never been in the Western Hemisphere or something like that. So you gotta be careful about where you're getting information. The pathogen must be pathogenic. That means we need to do Koch's postulates. We need to be able to kill stuff with it, right? There are pathogens in the textbook that can't possibly do anything, right? Like somebody in 1948 in a greenhouse somewhere killed something and it got in the textbook, and they're famous, so we're too embarrassed to kick them out of the book or something, you know? And so it's like they're secondary organisms, and it's good to know about them because they grow on dead plants and it signals some other problem, but that's not a primary issue, right? So you got to be careful about those. And then the pathogen's got to be virulent. Now, virulence is defined in different ways, right? I define it as efficiency. How efficient is the pathogen at doing its job, right? Like just use COVID, right? Omicron is more virulent than the other ones because it spreads easier. It's like 50% more transmissible, right? It doesn't necessarily kill you better, but it spreads better. And the idea for a virus is just to spread. And all living organisms, they just want to reproduce, right? That's their goal, to move the, like if they have a goal. I mean, I'm giving human characteristics. I'm anthropomorphizing it here, but you know, it's the way people understand it, and like passing along the genetic material. So if you're efficient at that, that means virulence, right? Now, some folks will talk about it in terms of damage to the host, but I don't see it that way. That's a genetic influence, right? Omicron had 15 gene mutations that made it more transmissible, right? I just read an article in Nature that says there's 2,100 more possibilities for that. Yeah, Yeah, I know, right? So we'll be zooming next year or something. Or we'll get a shot every 3 months. I don't know. Man. I caught it from a 7-year-old on Christmas Day. Yeah, my brother's granddaughter is like, or grandson, he's like, you don't look good. Oh, he got 102 fever while I'm sitting there playing with him. It's like, yeah, okay, here we go. And so I didn't really get sick, but it's still anyway. So it's a genetic thing. There's a genetic thing that runs it. We also have an environmental factor here. A lot of these pathogens are switch hitters, right? They go to sleep or they're saprophytes, and it takes a certain environmental parameter to turn them into a parasite, right? And usually, it's for fungi, it's moisture turns them on, and temperature is like the throttle, drives it. So there's that too. So like, if you're looking at a problem, can you actually have the problem you think you have? Can it cause the disease? Can it do the damage to the plant that you're looking at? And how bad is is it widespread? Is it doing a lot of damage? These are questions that you need to ask yourself when you're making decisions, right? But be careful about where you get information, right? I think that's, we all know that there's a lot of strange stuff out there on the interweb. And like I used to do Twitter. I signed up for Twitter for my job. And I had all these golf course superintendents. It was a marketing tool for me. And I got to the point where I couldn't look at it anymore because there was so much BS coming across that it would just make me mad. I'd look at Twitter for half an hour and then I'd be pissed off. I'm like, so I just stopped looking, right? So be very careful. If it says .edu, or .gov, then I give it more credibility than .net or .com, right? So again, be careful about that. All right, I keep driving here. Hour and I got about 10 minutes, and we'll take a short break, and then it's an hour or so. And so environmental condition, right? Provides pathogen opportunities, influences the host plant, increases pathogen virulence, right? Turns them on. And so we have this host plant condition, we get a more susceptible plant and we get a more virulent pathogen, and that leads to disease. And we call this predisposition, right? Site weather management. And certain these circumstances play a strong role in disease outcomes. And it's sort of like this, right? It's the fuse that drives the relationship. You got a host plant, maybe it's drought stressed and it's weak, it's respiring, using up too much carbohydrate, and then you start to water, and then the pathogen turns on, and bam, you have some negative impact on the host, positive impact on the pathogen, you get disease. So when you're looking at diseases, we're always looking back here for the circumstances that might have turned it on, right? And so that's an important thing to keep in mind. Those circumstances play a really, really strong role in disease outcomes. And so, a lot of these pathogens, we don't get them every year, but we get them under certain conditions. And are the conditions being met? Because I look at plants and sometimes I find more than one pathogen. In a turfgrass plug, I'll find 3 or 4 potential pathogens. And then I got to ask myself, well, which one caused the damage? And so we look at circumstantial evidence, right? And that point is there. Now, the last concept here is about how do you know when you got a dead plant? Because some people, they just don't want to admit it. I'll just water it again. Yeah, we'll just keep watering it, and we'll resurrect that dead thing. And so, that's not going to happen. And so, symptoms are the deviation from normal. It's the recognition or the observable condition of abnormal physiology. The plant's brown, that's a symptom. And so when you think about plants, you think about symptoms, right? Now, the sign is the physical presence of the cause. So when you're thinking about signs, you're thinking about like the mushroom, the fungal fruiting body, the mycelium, right? So what we want to try to do is put these two things together. Now, I put clear evidence of abiotic stress in there too, as a sign. It's 100 degrees on Tuesday and everything's brown on Saturday, cause and effect. So that to me is a sign. And so I just did a sample where a guy had some arborvitaes on one side of them, they were all burnt. and another landscape contractor had been on the property and sprayed everything with deer repellent, right? And the deer repellent probably burnt them because there was a cause and effect. So that the fact that the guy was there like 7 days before the damage and there was a pattern, it was pointing toward the deer repellent, and so I kind of, it's loose, but I call that a sign too. And so all right, so symptoms are stuff like this, right? And look at all the terminology that nobody ever understands. I have no idea what enation means. There's a couple of terms up there. I'm like, what? And like one day I got a plant pathology book, just made a list and then made the slide, And so the esoteric terminology, don't let it scare you, right? Just remember you're describing the plant. And if it has a spot, call it a spot, and like, yeah, if the leaves fall off, just say the leaves fall, there's multiple terminologies for a lot of this stuff. Like if I saw that, I'd say it was blighted, the tissue was blighted. But you'll see it called dieback, you could see it called flagging, so just keep in mind that you're describing the plant. What we're trying to do is just describe as best you can what you see, and so, some of these terms have a little bit of meaning to it. Like if you tell me I see a patch of dead grass, I think it's round, right? Not just a big irregular area, because we call diseases in turf like brown patch or summer patch, and they're rounds, fungal diseases, right? So it means something to me, but still, if you said I have a patch of dead grass, I would say, is it round? And you would say no, and then I know it's not a patch, right? So I can just forget about brown patch and summer patch, right? And so insects do it too, right? Insect feeding and breeding behavior. You pull the bark off, you see something like that, that's a bark beetle. And then you go, what bark beetles on pine? All the insects in that same family have some sort of similar symptom that relates to how they eat the plant or they breed in the plant. So you can think about that too. You got that with pesticides, right? Herbicides. This is a material that was taken up by the plant, moved out to the tips and burnt all the tips, right? And so symptoms, there's a symptom, there's a consequence of all the different causes that will help us tell what cause it is, right? So I knew when I saw this that it was a growth regulator type herbicide, because they move in the plants to the tips and high concentrations will burn the tips. But look how it's all twisted up. These materials are auxin mimics, and that's what they do. They twist. So a low dose will just twist up the plant, and a high dose twists it up and then burns it, right? So as soon as I saw it, I knew what I was dealing with, right? I didn't know which herbicide or what exactly happened, but I knew that that's what it was because it was a typical symptom. And we'll get to that, like typical symptoms of fungi, bacteria, virus, and stuff like that. So you look at symptoms, and what we try to do is describe them and add them together. Symptom A, symptom B, symptom C, symptom D, and now you have aster yellows. And you can see that it's yellow. It's, you can't tell it's stunted, but look at all the shoots compared to the healthy one, right? And then this is what the flowers do. You can see it down here. That's called phyllody, and that's pointing you toward a disease. Right. And now you can make a pretty good educated guess from this and go, that looks like aster yellows. And now that's when it comes into me and you say, well, it looks like aster yellows. And I'll say, okay, it looks like aster yellows, and it's going to cost $272 to treat or to test. But I'm going to prove that it's aster yellows. I can prove that it's a mycoplasma, but the protocol costs this much. Do you want proof or do you want to guess? Because the guess is probably about 80% certainty. And if I run an antibody kit or if I get a PCR, we're at 95% certainty. Do you need that certainty? And like, a nursery grower, over at Centerton or something needs that certainty, right? But a homeowner probably doesn't, because the nursery grower has a cost, right? The homeowner is just going to yank it out and get a new one. You know what I'm saying? So maybe that's good enough just to go, that looks like a yellows disease, probably aster yellows. So that's what I want to try to promote the idea that we all can solve problems, even if we don't have the same tools that I have in my lab, right? Because we're trying to solve problems. We're not doing research, right? So sometimes close enough is good enough, right? Is that true? Horseshoes and hand grenades, right? Yeah, that's what my dad would say, So like, look at this, like symptoms are dangerous though. So you see the fuchsia, it's wilting. So I go around the room, what causes wilting? You know, everybody says not enough water, and that's true. But if you water too much, it's going to wilt too, because the roots don't work, right? And like, if you water just enough and the evapotranspiration demand is high, it's going to wilt because the water's pulling out. You know, if you put too much fertilizer and the roots get salty, water's going to move out instead of move in, right? So it's going to wilt. Or like say an insect chewed the roots, it's going to wilt. Or a fungus rotted the crown, it's going to wilt. Or if there's a bacteria in the xylem, it's going to wilt. But wilt, you know, signals moisture stress. But the cause could be anything. If you assume that it's water and you water it, you can turn that fungus on, and now you got a bunch of dead ones, right? So you've got to be careful. It's not the end, it's the beginning of solving the problem, right? So we need to recognize all this stuff and then start making some assumptions and then collect more information. And what we're trying to do is get to the sign. Like if we can get to the sign, and all this except for this insect stuff in the middle says mushroom, right? We're looking for fruiting bodies and mycelial characteristics and spores, right? And we're looking for bacteria. And sometimes signs are easy, like cedar apple rust, right? Once you see this, like you'll never not see this again. You know, it's Gymnosporangium juniperus virginiana, right? And eastern red cedars are all over the place as weeds. And so seriously, around the first week of May, start paying attention. They're about the size of a baseball, right? And they'll be once you see it, you'll see them all over the place. Here's a tree in my neighborhood. You know, that's, yeah, it's awesome. In May, it looks like Christmas, right? Yeah, it's really cool. And it's not going to kill the tree. But no other fungus looks quite like this, you know? And so once you see it, you know. And the problem with these signs, though, is most of them look like this. You know, it's on this twig and there's a little tiny black dot. And what is it? You know, that could be a parathece or a pycnidia or a acerbulus or a sporodoc. You know, I'm going to know because I'm going to section it and put it under a microscope slide. And I'll be able to tell what the structure is and see what the spores look like and name it. Right now, look, that doesn't mean that you seeing this with a hand lens in the office over here, you know, isn't valuable information, right? You see a dogwood and you got twig dieback, and you go, well, dogwoods are going to die back because it's hot and dry, but they'll also die back because they get dogwood anthracnose. And you look really close at the dead twig and you see the fungus. You go, maybe that's dogwood anthracnose. And then you tell the client, you know, with dogwood anthracnose, the best thing to do is prune off the dead branches. So you teach them how to prune, and then maybe you should water your dogwood, right? You know, and that sort of thing. And you solve their problem. You know, if it's a nursery, I got to see this because I'm going to be recommending the pruning and the watering, but I'm also going to be recommending a fungicide protocol. And the fungicide protocol is something that you can't generally recommend to a homeowner because they can't buy the material or spray it right. Right. So what's the point? You know, you might recommend so that you can help them hire a professional to come do it, you know, but then that's a subjective decision about how valuable that tree is and all that sort of stuff. You know, so again, you know, I'm trying to build on this idea that you could solve problems even if you can't get to this part, right? And so again, you want to be careful about signs. They're not always the end because of the fungi that are imposters. And like this one, these are pictures out of the Compendium of Rhododendron Diseases, the American Phytopathological Society. And Pathologiopsis causes this disease of rhododendrons, gray leaf blight. And so when you read the commentary by the pictures, you find out that this fungus gets on plants that are in this condition, you know, that are winter damaged or drought stressed or something. It can't kill anything. It jumps on in a secondary way. So if I'm looking at it and I see it's Pathologiopsis, then we back up to the horticultural problem, the winter injury or the moisture regime or something like that, right? So you want to be careful. All right, last thing here, disease cycle, infectious diseases cycle, right? And I'll just do this quick. I won't talk about apple scab that much, but here's a pathogen. It's causing the scabby lesions on the leaves, and those lesions have fungi growing in them, right? So every time a raindrop hits it, it spews the spores. And those spores continually reinfect leaves. And as the season progresses, the trees drop all their leaves, right? So most crab apples by the end of the seasons are pretty scrubby looking, right? With no leaves. Now, the thing that happens then is the pathogen gets in the fruits and the fruits fall on the ground and they make another structure called a parathecia that overwinters and starts the disease up the next spring. So when you look at that in a disease cycle, it looks like this, right? So what do you do by knowing this? You know, if we understand this, it gives us insight in how to control things, right? Maybe we rake up the leaves and the fruit so we don't get this part, right? Or maybe we spray a fungicide here so we don't get the initial infection, or we spray like through the season to protect the leaves, right? So this is the thing that you know, when we see diseases in an office or in the lab, it might not be the right time to control them. So we go back to these disease cycles and then we say to solve this problem next year, you know, you have to spray this and this and this at this time and that sort of thing. So again, you know, just be aware that there's a time associated with all of this. And again, I'm not going to do these last couple slides. All right, so let's take like 5-10 minutes. Can you guys and sit back down, go, you know, and we'll start back up. don't have a specific. But there's a company called Gempler's that sells a bunch of different ones. Our soils guy, Joe Heckman, tested a bunch of different ones, and all the cheap ones work just as good as the real expensive. Oh, wow, really? Yeah, so if you spend $50 or something, like mine costs $700. One in my lab, and so if anyone, we're going to work on that. So, yeah, Gempler's. Yeah, GMPLE, and they sell all kinds of stuff, like everything from like shovels to pesticides. It's what a lot of the professional landscape guys. That's a wholesaler. So yeah, and I know you can buy off. Yeah, they got a website. I know. Yeah, I know you can buy for sure. Thermo Fisher, they have a website, but that's a giant company that sells to sells to universities and stuff. You probably could buy from them, but it might be more difficult, more hoops to jump through. So, it doesn't really work. It works because you over fertilize. If you follow the label, you're putting out like 10 pounds of nitrogen per 1,000 square feet. And so you just wind up with a really dense lawn that's not going to have crabgrass anyway. Right, so it looks like it's working, but it has more to do with the fertilizer than like the herbicidal effect. So like you can spend a lot less money to find urea and fertilize your lawn bed and get the same outcome. So that's kind of, it's been around for a while, and there's a lot of great efficacy, but the reason it works is it has more to do with fertilizer.
And it depends what credits you need.
So yeah, be careful about that.
So
It ain't
for
cheap.
the private applicator.
All right.
Whatever says
Yeah,
PP2
yeah,
on
and
it.
urea is cheap,
PP2.
Mhm.
so,
Yeah, so. and there-
all right, class,
Okay.
Yeah,
And what does
Class.
PP2 mean?
that's private. Yeah.
Okay.
okay. Yeah, that's all right, I understand, and I'm slow. All right, you guys ready? Oh, no. All right, here we go, part 2. Now you're plant pathologists. Yeah, right, everybody's trained and ready to go. So, Mary can go home, take care of her chickens,
Right.
and you guys can do all the work. So,
That's the plan.
yeah, that's the plan, right? And so anyway, all right. So what are you going to do? Somebody gives you a dead twig and says, "What killed my plant?" And so that's what this part's about. And so try to go as fast as I can so we can get. I should be able. I'll be close to noon. And
7.5.
theoretically, I could do this in an hour. Identify the plant. Step 1. What's the plant? In my world, that's the hardest thing to do. And you'll see it because people come with a dead branch, or one leaf they picked off a chef Lair or something, what's wrong with this? And like, I have no idea because I have no idea what plant that is. And once I think this is the most powerful part of this. Once you know what plants you have, then you understand what's supposed to go right for that particular plant, right? And so once you know what's supposed to go right, then you get a clear picture of what's going wrong, and I think that's an important thing, this horticultural requirement. And in the example, I'm going to use a spruce, right? They call spruce Colorado blue spruce and Norway spruce. Where do you think those plants want to live? Right? Is it Boulder, Colorado, or like Tom's River, New Jersey? I know where I want to live, and so so there's an inherent problem when you get these plants outside of their normal range. It's hard to meet their requirements, so they're going to have problems. and sometimes just by knowing what plant it is and seeing where it's planted, you can tell why it's dead because of the abiotic stresses that it's faced with, right? So that's an important thing right there. It's how we rule out the abiotic, or how we start to see the predisposition, the predisposing factors, the things that are making it weak enough to get disease. The other thing that we get once we know what it is a list of pathogens, right? And so everything has its own list. Not all disease get in all plants, not all plants get all diseases. And so we get this list, and then on the list, there's always a main problem. So in integrated pest management talk, you'll hear about something like key plant, key pest, key location, or something like that. And my definition of that is main plant, main problem. Every plant has a main problem, and so what we find in the laboratory is we get the main problem over and over and over again. So, you rule out the abiotic stress first based on your clientele. Uh-oh. They're not seeing that. I'm sharing the screen. See, they should have showed up. All you did was, you guys should have came. Yeah, I got my shots and I had my infection, so I'm good.
They told me to reset the screen
I
share.
went to 20 concerts between July and October last year. And I've been to 7 so far this year. So far, so good, right? A lot of music, yeah, live music is, it's my hobby, so. And yeah, I got tickets to 4 coming up. So anyway, who? Going to the Who? No, I've seen them twice already. I got Jason Isbell, and I got Hot Tuna, I got Wilco. I just saw the Tedeschi Trucks Band 3 times in Red Bank. You know them? Everybody knows them? They're good. If you like the Allman Brothers or Eric Clapton or something, that's Susan Tedeschi can really sing, and they can play. Anyway, so, but yeah, that's what I do for fun. And then I ride my bicycle, which I'm going to do this afternoon. Because we get it done here at 12, I can be home 1:30. What's the point of going to work, That's the best part about the pandemic and the Zoom thing. Nobody knows where you are. So, as long as the work gets done, it doesn't matter how or when or what. So anyway, all right, so here we go, right? So here's the spruce, right? And so in my lab, we have data, we collect data, and so I had a grad student a couple years ago, Mary Babiez, and I said to her, let's ask some questions of the database for her thesis. So she just mined our data for all this information. And what we found out was that oak trees are the most common plants submitted to the lab. And that's kind of weird because oaks are pretty sturdy. And the reason why we get oaks is because of a disease bacterial leaf scorch. And we work on research projects with bacterial leaf scorch. So I'm getting trees from the Forest Service and the State Department of Ag, and that skews the data, and so if we ignore that, the second most common plant is boxwood, right? And then the third most common plant is rhododendron. And then the fourth most common plant is spruce. And spruce has been coming up the list. Boxwood and spruce have been moving up. And then it's stuff like, maple and pine and pear and cherry and, a lot of stuff that everybody's growing. And I always wonder, like, are we getting these plants because they're the most problematic? Are we getting these plants because everybody has them, right? And I think that it's more of the second than the first. But, if you're using a plant that has a disease that's pretty common, it's going to run up the list because we're going to get that disease. And with spruce now, all of them have this fungus on them called Rhizosphaera. Right. And so Rhizosphaera is not a really virulent pathogen. It takes a little bit of stress to get that pathogen in the trees. But once it gets in the trees, it knocks the needles off. It'll kill them. right? And so we get spruce and they use them like this, what's that guy, K. Hovnanian, built a bunch of neighborhoods and got a bunch of cheap spruce trees and planted them like this. And that's a heat sink and that's a drought stress area. And look, spruce handled drought pretty well, but not that good, not as good as the book said. They handle wet sites even pretty well, but not that good, not as good. So you get on the extremes of moisture, and we see like really wet sites stressing them, and we see dry sites like this stressing them, and so they're going to drop needles anyway. We already have them coming in the lab because of winter damage. This winter is not a good year. It's too warm. So it's warm, it's 70 and then it's 22, and so the trees don't acclimate properly, and during the cold, they burn up. And so we're already starting to see it. Tuesday I was in the lab, or it was Monday, and it was all spruce and arborvitae and all winter damage, right? And so we get a little winter damage, and then Rhizosphaera comes in, right? And so that's the key pest right now in spruce. And so this tree had it, but again, they might look like this anyway because they're on this site. And then you look at the needles and you see the fungus, and then the fungus drops the needles off, and then the trees fail. So there's two things here, right? Colorado blue or Norway spruce, it's not growing in Norway. You put it on a berm that doesn't get any water on a crummy, construction site, and it's going to fail that way. And then it's got a key pest that's going to come in and kill it, right? So it's an issue. Like I'm an anti-conifer guy. I'm not planting any of that stuff, because if you plant Doug firs, you got needle cast problems. Now you plant spruce, got needle cast problems. You plant the 2 and 3 needle pines, you got borer problems, you got a bunch of stuff problems there. And then you got all the minor ones, Chamaecyparis and Cryptomeria and all that stuff. And then you come back, all you got left is Eastern red cedar, which is a weed, and like nobody wants that. My grandmother used to want one for a Christmas tree, right? Which I could never figure that out. My Uncle Bill would go out and cut one down, and he'd hump it from the woods somewhere, but I guess that's just from the Depression or something, And so, but again, we have these plants that have problems because they're planted outside their center of origin, and then all of them have a key pest, right? So how do you find out about this stuff? So here's a book, Manual of Avoiding Landscape Plants by Michael Durr. Michael Durr is at the University of Georgia. And he's the preeminent horticulturalist. And I like Michael Durr's books a lot because what he does is editorialize. He says what he thinks. And that's not quite proper for these kinds of books, but it's valuable information, right? And so you got to know what you're looking at when you look it up. And it talks about all the horticultural requirements, everything is supposed to go right. And then Durr talks about how to grow it and how to use it in the landscape. He always has a big commentary of all the varieties and stuff. And it's interesting because he'll tell you what's supposed to go wrong with the plants. Like purple leaf plums, they're blooming now. Those things are 15 years. Durr says it, this is a temporary investment, or Leyland cypress, temporary investment in your landscape. You got 15 to 20 years and these things are going to start to fail. And so I'll get a sample and I'll say, well, when did you plant it? And it's 22 years in the ground. And I'm like, well, you did good, get a new one. It's time for it to die. And so you got to recognize that's going to happen. And that's what I find out. I plagiarize this book all the time into my letters, and if there's an argument with landscapers and stuff, all I got to do is bring up Michael Durr. Everybody respects his opinion. He's the one that wins, so I just put according to Michael Durr into the letter and then he wins. Now, the other thing that Durr does for you is gives you a list of problems, right? And this is a layman's list. This is the key pest list, not a, there's a book called Fungi on Plant Products in the United States. And if you looked up maple, it's like 10 pages of tiny, like one of those giant dictionaries in the library you can't pick up. And so, you look on the list and now you got something, you got the abiotic stress and the horticultural issues and the key pest list. So this is the valuable textbook. I use it all the time. Because of that, step one. Now, step two, we're going to start looking at plants for symptoms. And we're going to start making decisions about what's going wrong based on symptoms. So the first thing is what plant part, and then is it abiotic or biotic, right? And then is it fungus, bacteria, virus, something like that, right? So we're going to start with identify the entire plant. You got to look at the entire plant to tell what's going wrong. Spectacular abnormalities in the top sometimes don't have anything to do with the top. They have everything to do with the bottom. So this is a sample that came in. The guy brought 5 of them to the lab, and our rule of thumb is don't say anything until you swipe the credit card. And so so I already knew, it's from a nursery. I already knew getting them out of the truck what the problem was. And the guy drives off, and I yank it out of the pot, and in like 40 seconds, find this, right? It's right underneath the soil line. All I had to do was yank it out of the pot, right? Yank it out of the pot, dig it up, yank it out of the ground so you can look at the roots. I can't. I'm of the opinion sacrifice one for the good of the many, right? If you let it go, it could just get worse and cause more problems. So if you want a branch like this, maybe when it's starting to look like this, you prune some of these out, you cut them up. If you wait until it's like this, then when you're cutting on the main stem. And so you see the canker here, right? And so, but you got to be able to cut the branch in order to see that. So if you cut the branch and there's no discoloration, it's not going to hurt the tree. It's going to compartmentalize over, but you got to cut it. You got to be willing to cut it. And I think that's the point, because what you need to do is define what plant part is dysfunctional, right? And so with most fungi and most insects, they attack specific plant parts. So if we can figure out what kind of disease it is and what plant part or physiological process is disrupted, then we can narrow down that list, right? And so Cynthia Westcott is, probably the first great woman plant pathologist in the 40s and 50s, wrote this book. She was at Cornell, and it's interesting because the editor, Ken Horst, is dead now too. It's six versions in. And so this book's been around a long time, but what it was she made a list and then listed all the diseases by kind of disease or plant part, right? So you look up, so you define the problem here as a canker, and you look up the tree and you look canker, and now your list just got smaller, right? And the one thing about Westcott's book is, you see the states, right? Those states are from a handbook, an index of plant diseases in the United States that comes from the USDA. Right, and that's just a list of frequently encountered pathogens in the United States, and I contribute to that list for New Jersey, right? And so you got these, and so you're looking at something, and it says North Dakota, South Dakota. It's probably not the one you're finding here. It's probably the one that says New York, Vermont. It might be, but again, it's circumstantial evidence that helps you narrow down your focus. So Westcott's a good book for a couple of things. Like, if you think it's a disease, you look up Westcott and it's on the list, you're probably right? Or if you can just say it's a canker, which one of these fungi, and that she gives you a short list and that sort of thing. So it's a valuable text. There's an insect companion to it. This book is not cheap. If you buy a brand new one, I think mine costs $2.90 or something. Yeah, so if you go find a used one. If you see a used one for $8, buy it. Right, and so that's my mom gave me this book when I got my master's degree, and for my gift, and instead of my check. And so she's like, I already gave you too much money to get it. Anyway, so that's kind of evaluating the entire plant. You need to see the entire plant, and that's the best situation. You can always plant it back. Think about that. If it's a rhododendron or something small, dig it up, look at, maybe you need to turn it around anyway, or recite it. So evaluate the plant community. So each plant family has similar problems. So this is where we start to look at abiotic versus biotic, right? So if you look at a landscape like this, and this is what passes for a corporate site in New Jersey. It's got all the requisite plants. You got the spruce, you got the pear, you got the miscanthus, you got the euonymus, right? You got the tall fescue sod. Everything looks like this, right? And so if you look at the landscape and you see some sort of symptom on everything, it's probably not a disease, right? It's cold temperature, it's drought, or it's something that got sprayed on the grass, it drifted on everything, right? You know, even if it's drought, all the different plants will exhibit drought stress a little bit differently, but the symptom that you see will be in the window of all the possible drought stress symptoms, right? So you know, some tolerate better than others. But if you come in the landscape and you see the miscanthus and the tall fescue, both in the Poaceae, it's likely to be a disease, right? Because you know, not all plants get all diseases, and the similar plants get similar problems. Right, so you're looking at the landscape like that, and you're narrowing down focus, and so you know, it's a difficult thing to do, because if you're helping somebody in the office, you can't go to their site, right? If I go to someone's site, they're paying me $1,500 just to get out of the car, right? So like, no homeowner's going to hire us to come, you know, and and we go and that sort of stuff, but like, it's you can ask a lot of questions, you know, what's everything else look like, you know, or, you know, the good thing is that everybody's carrying around the camera now, you know, or if that's a bad thing, maybe, you know, when you're hanging out with my 22-year-old who's got to like Instagram every 5 minutes while we're hiking in Colorado, you know, it's like, but you know, it's, did you take a picture? Right, we try to get all of our landscape guys to give us pictures, you know, so I got guys coming into the lab with their samples and airdropping the pictures right to our computers while they're standing in the office, and so that brings you to the site, and that means you can see the landscape. So that's, you know, I tell the golf guys, you could pay the consultant $1,500, or you could just send me a sample and a picture, and I'm standing on your putting green with you. You know, and so pictures are good. Now, the other thing that you can get too here is this, right? Remember how I said that every time I go outside, I'm at work, right? If you get that mindset, if you're mindful about what's going on and you're looking at stuff, right? If you walk out of your front door and you see something going wrong with your rhododendron, I'm telling you, it's coming into the office here. You know, the same problems. We call it the disease of the week. We get the same kind of problems from the same plants at the same time every year when their key pest flares up, right? And I recognize it, you know, and I see it all over the place. And so start thinking in those terms, you know, in your daily travels, just pay attention, and then you'll see what's going to happen. You know, it's easy to predict. So again, looking at the communities with that kind of an eye helps you make decisions, right? Then we start looking for things like patterns, right? So patterns relate to environmental and site and cultural inputs. Uniformity is usually abiotic, and randomness is usually biotic, right? So you look at something like this. This is a nursery I ride my bicycle by all the time, and I'm always taking pictures. And I've had the guy like, I'm glad you're not telling anyone, you know, who it is. But look at these boxwoods. See every one of them. You know, and dyed is a little melodramatic. They're just brown from the winter. Look at that, and every one of them is brown. And look at this variety here that tolerates winter better. Every one of them. So there's two patterns, every one of them, and there's two different varieties that are acting two different ways, right? And so you think about that as an abiotic stress. These could have been sprayed and burnt, or these are getting watered, these are not. You know, the proportioners for the irrigation system, these got too much fertilizer and burnt up, you know, stuff like that, you know. And so you compare it to this, you know, it's a Christmas tree field, right? And there's a disease called Swiss needle cast. And depending on where you get your seeds or where you get your seedlings from, you're going to either have rhabdocline needle cast or Swiss needle cast. And with Doug firs, it's about where they collected the seeds originally, what national forest. And so in the Rocky Mountains, there's shuswaps from British Columbia and northern Rocky Mountains. And then there's Lincolns from Lincoln National Forest in New Mexico and Colorado. You got Lincolns, right? and Lincoln's get rhabdocline. So we started telling everybody to try to get shoe swaps. And in a choosing cut, right, you just replace them randomly. So this guy, Steve Willard, he was starting to use shoe swaps. And we realized that that was a mistake pretty quickly because the shoe swaps get Swiss needle cast. And this was the first picture of that. And all the shoe swaps are getting, the random trees that were replaced randomly got the other disease, right? And so it shows up like that, and the Lincolns are all holding up, right? So again, just like you see the pattern and you see the randomness disease, pattern, abiotic. Now you got to look at individual plants too. Like you see drought stress looks like this, every leaf nice and even, every leaf in the canopy, and you compare it to bacterial leaf scorch. And it looks like drought stress, and it is a form of drought stress. But look, this one's totally stressed, this one's not, right? So it's irregular, it's subtle, but it's something that you got to keep in mind. And so it could be plant community, it could be individual plants, could be individual plant part, but you'll see the patterns. I had it was down here in Cumberland County, and I had a nursery guy brought me a bunch of trees that had spots on them, and they were all different trees, and they had these spots, and they were all the same size. And I was like, asking him questions and stuff, and what did you do? And we didn't have a good answer. And then he said that most of these trees are along the road, and I was like, And then so I called the county agent, and I said, what's going on this road? And I'm like, is there a farm or anything, or what's going on? And he says there's a farm across that's growing small grains. And I said, go find out what they sprayed. And they went, and he went there and asked them some questions, and they had sprayed diquat, right? And diquat is an herbicide, and it drifted, and it burns by contact. So it had drifted over onto the nursery and burnt all these trees. And like the reason we started asking those questions is because all the spots were the same size, so there was a pattern, and then it was across a bunch of different plants that weren't related at all, so it couldn't have been a disease. And then when the guy said it was by the road, there's another pattern, so like you start using these patterns, and they'll send you down different paths of inquiry, looking for the circumstances, the circumstantial evidence. So that's kind of cool, that sort of stuff. And so then we look at symptom progressions, right? Symptom progressions indicate living or non-living, right? So if it's progressive, we all had chicken pox, right? We're all old enough to have had chicken pox. And so you get a fever, and then you get a rash, and then your sister gets it, right? And then every kid in the preschool got to stay home, and so it's I think I got it when I was in kindergarten. Everybody was home, all 80 of us or whatever. So you compare that to crash your bicycle, break your arm, and I did that too. And so like you see something like this, right? This is the failure of rhododendron. So this was I took this picture in 1990, believe it or not. And I was a grad student, and we had 360 rhododendrons. And my job was to inoculate them all with Phytophthora, and then spray different fungicides at different fungicide rates and wetting agents. We had all these protocols. And I don't know what I did, but I killed them all. So I had to go out there every Monday and rate them 5, 4, 3 for statistical analysis and this was the rating system, 5, 4, 3, 2, 1. So all that work, the only thing we got out of it was this picture, but it makes a good example about how Phytophthora kills rhododendrons. It was about 8 weeks from inoculation to death, and so you want to think about this in terms of time, right? Bacterial leaf scorch in that last slide, that takes 10 years to kill a tree, right? So boxwood blight will take a week to kill the boxwood, so like that time though, and the idea that it's spreading is important. Like think about this as a foundation planting, and this one dies, and then this one dies, and then it spread into this one. I always get the comment where I planted a rhododendron, it died, and then I planted some hollies, they died, then I planted a forsythia, it died. When someone says that, that's Phytophthora. because Phytophthora is one of the exceptions that gets on, right? You know, or it died and it keeps spreading. It's going right down the road, you know, something like that. So, you know, if you're looking at that movement and time, think living organism, right? Now, this is glyphosate vandalism. And yeah, somebody on the crew poured Roundup in the tank, and they sprayed 10-acre corporate site with it. And they didn't think it was very funny. I got on the site. I said, didn't look like you got good coverage, because all these areas they were using one of those guns on a hose, and they didn't get good coverage. And it took about 2 weeks from the application for it to die. And you should see that. I got pictures. It's crazy. And so cause and effect, and it's quick. You know, it was still 2 weeks, but 2 weeks is quick compared to a disease. And so again, I hope that illustrates. You know, glyphosate. We get a lot of like, my neighbor killed my plant stuff. And sometimes it's true, and every time it's glyphosate. Every time it's glyphosate. I had a guy over in Egg Harbor, I think, looking out onto the water, and like he couldn't see the water through his neighbor's arborvitae, so he killed the two on the end. And the arborvitae, the dude was like some high-powered lawyer in Philly. I'm like, you killed the wrong guy's plants. You know, we were getting subpoenas anyway, but it's always glyphosate. You know, I'll tell you this too. I got great pictures of Trump Bedminster's golf courses, where somebody after he got elected went on the golf course with a sprayer and wrote stuff on the putting greens like tiny hands and stuff. Regardless of your politics, it's hilarious looking at. Like who thought to do this, you know? And I can't show you because some of them are foul, but you know, it was just like, it's always glyphosate. It's always glyphosate. So anyway, when it's vandalism, and like we do about a half a dozen a year, and so all right, so kind of get a feel for that, like how to do abiotic versus biotic and how to think about the entire plant, right? So now what do you got to do? Like, is it bacteria, virus, or fungus? You know, or is it an insect? So what about specific kinds of symptoms from each of these different problems, right? So we start with fungi. Fungi cause most plant diseases and attack all plant parts, but there's a commonality about this. They usually transmit by spores. And so there's a central infection point where that spore germinates and the germ tube invades the plant. And then the fungus grows in concentric circles. So on the plant, it looks like a round circle, right? Now we talk about how fungi absorb nutrients from between the cells and makes a dry rot, right? So we got these dry circles. Very often they have a discolored border, and fungi make signs. They reproduce pretty readily. So we look at something like this, zone 8 leaf spot, and it's nice and round, a nice even border. Look at this discolored margin. See how dry and papery it is? You can even see some concentric circles down here, right? And so that's classic fungal disease, right? And again, it's on a leaf, but you can still see similar things if you're looking at a branch or woody tissues. And then your eye is drawn here, right? Because it's a cherry and it's bleeding. It's not soft and slimy. That's just what cherries do. But that tells us we need to look here, and we cut the bark away, and you got a nice and round, even lesion, you know, a nice dry rot, discolored, you know, and so even on the wood, it sort of matches up the parameters that we have. In every case, it's similar, or we have most of those characteristics. So here's another one, right? It's not round, but it's dry and papery with a distinct margin, a colored margin, right? The folks that sent this to us, they thought this was transplant stress. And the transplant stress is usually a drought type reaction. So we thought we would have saw a pattern, you know, the tips and the edges of all the leaves, but it wasn't. It was a random pattern. So we assumed fungi. Anthracnose diseases cause angular spots rather than round spots. So we assumed it was an anthracnose. In order to prove it, we took the leaf and we put it in a plastic bag with a wet paper towel. And 24 hours later, we got the fungus fruiting on the bottom, right? And so I just put this under the microscope and I named it aerial basidium, right? But you'll look at this and go, there's the fungus. So that's probably an anthracnose. And, you know, what do we do about anthracnosis in maple? You know, something like that. Maybe Ohio State has a fact sheet or something like that. You know, Rutgers doesn't have a fact sheet, you know, but there's information sheets from land grants that you can hand to your clients or whatever. And so again, you know, this idea of dry and papery and discolored and signs holds up pretty well for most fungal diseases. So you compare that with a bacterial pathogen. So bacteria are on the plants all the time, and they get into the plants through natural openings or wounds. Now, most of the bacteria that we see are on herbaceous plants, right? I'll get bacteria. I get a bunch of like cabbages and stuff from growers down here growing bok choy and that sort of thing. We suddenly have these clients that are growing stuff in warehouses, right? And they send me all kinds of bacterial problems. You know, and then it's hostas and zinnias and stuff like that. You know, a lot of perennials, all kinds of herbaceous stuff. Peppers, we had some pepper transplants in that had bacteria in them. And so you know, that's common in the more herbaceous stuff. There's a couple of exceptions, bacterial leaf scorch and fire blight. But so they get in angular borders versus round, chlorotic halo instead of a discolored margin, water soaking instead of dry, right? And tissue blight, rotten smell. So this heuchera, remember what's happening. These bacteria are making toxins and they're killing the cells, and then they break them apart so that they can absorb the nutrition, right? Using an enzyme. So you get this water soaking. It's easier to rot the cells in between the veins than it is the veins. So the spots are confined by the veins. So if it was corn, like Stewart's wilt, it streaks up between the veins of the monocot, right? And so you see the halo here. That's where the toxins are starting to leak, right? And so tomorrow, that's going to be black, and the halo is going to be bigger. And the next day, it's going to be bigger until the whole thing turns to slime. You know, fungi generally turn off or confined on their plant part. Bacteria, if you let them go, it's going to rot the thing to nothing, right? And so we see that sort of like this. This is fire blight, and the bacteria, Erwinia amylabra, lives in the tree after it's infected, and it'll ooze out lentils and stuff. That extracellular polysaccharide ooze. It's attractive to pollinators, and the bees and flies and stuff come get the bacteria when they eat, gets on their body. They go to pollinate the flower, and they infect the flower through the nectar cells, and we get a spur blight. This is really common, right? If we have the right weather conditions after the spur blight, which is usually cooler and wet, right? Then spur blight turns into a branch blight, right? And you get these shepherd's crooks and stuff. And so if it's right, it'll kill the whole tree. I mean, fire blight's a pretty damaging problem if you're trying to grow a pear commercially in New Jersey. You know, it's one of the reasons why we don't have commercial pear production. You know, we grow peaches, you know, and so apples and pears are troubling pears worse than apples. And so you see this on street trees all the time. But the degree of damage depends on the weather. It'll keep going if the weather's right. Like this primula, right? The primula, wet, water soaked, those things turn to slime in the lab, you know, and I went to the greenhouse and there's like 15,000 of them. And I walked in and the stench was overwhelming. I mean, it was awful. And like everybody knows the smell, right? You know, from that potato that got away. And I remember I had these onions with slip skin. It's when one of the bracts gets this bacteria, you can squeeze them and they go, you know, and my lab stunk for like 6 months after they were in there. And so that's the thing about bacteria: rotten, slimy, wet, stinking mess, right? You know, and most of the time it's something like this as opposed to the fire blight. You know, sometimes we can see signs with bacteria. With a fire blight infected apple, I could put it in a moist chamber and get some bacteria beaten up, but most of the time we can't, right? So you know, if you're incubating something and just turns to slime and nothing ever fuzzes up and grows on it, that's telling you something, right? How about viruses? Remember what they're doing, abnormal growth and abnormal color, right? So the plant's making viruses, it's not doing its thing. Now you have to recognize, and we all know about symptomless hosts and latent infections now, right? You know, asymptomatic. And so that happens in plants too. Sometimes, like rose mosaic, it takes 2 different viruses to get symptoms. So you have plants that are infected showing no symptoms. You get them together, and now they infect each other. Now you got symptoms. So they're kind of tricky in diagnostics. One of the things that you'll see is that a bunch of different viruses in the same plant, like say in cantaloupes or watermelons, there's 4 different viruses that get in them. They all look the same. You can't just look at it and go, oh, that's watermelon mosaic virus number 2. You can't do it because it all looks the same. There's other viruses that no matter what plant it gets in, the symptom's different, right? So it's hard to just look at a plant and say what virus it is. You can say that it's virus infected, but you can't say which one, right? And so this one's cucumber mosaic. And the reason I know is because we tested it, right? And Sabrina, my assistant, this is from her garden. She has a 60-foot by 100-foot garden. It's awesome. 100-foot rows of lima beans. Doc Martens, big ones. And so, I know people. I like people who appreciate lima beans. And so, like green potatoes. And so anyway, my Aunt Mary Ann would just boil them with a little oleo. She would say a little oleo, a little margarine, and drink the water too. Anyway, but I hated them when I was little, but they're like gold now. And so, but anyway, so it makes these ring spots. It looks a little fungus-like, looks a little bacteria-like, but you put it in a moist chamber, nothing's ever going to grow. It's never going to turn to slime. It's only just going to have those rings. And I'm pretty sure Sabrina ate that pepper because, it was she eats anything. And so, as long as it's a plant. And so nothing can happen. Here's a hosta virus X. Think about this one. Look at this picture and think about what you're going to do when you're in the nursery and you see that and you go, oh, that was different. I'll buy that one. Because there was named varieties on the market with hosta virus X at the time. So again, the symptom doesn't have to be a ring. It could be just stunted plants. It could be modeling like this. It could be twisted leaves. Here's the hosta with impatiens necrotic spot. It's got rings. And like this one probably won't kill it, but this one probably will, right? So you got different outcomes from these different viruses. And so impatiens necrotic spot's one of them ones that gets in like 600 plants. And no matter what plant you get it in, it looks different, right? Like this is what it looks like on the impatiens. Almost looks like a fungal disease, but you see these stunted leaves. And here it is on coleus with black necrotic spots. So again, symptoms are helpful, and it's going to be some kind of growth or color issue, but it's hard to tell which virus it is, right? And so one of the things you want to recognize too is that pesticides and fertilizers sometimes look like virus problems. But pesticides and fertilizers have an applicator, right? Viruses have a vector. And so if you find a lot of insects, think virus. If you find a lot of nothing and see some patterns, then think chemicals, right? Something like that. Now, the only thing we can do with viruses, when we test them, we use antibody-based test kits, right? The Binax test kits. It's interesting because when I had Omicron, I thought I had it, right? And I called up Sabrina and I'm like, you got a test kit? And she's like, yeah. And she says, I'll meet you at the Tractor Supply. And she's like throwing it to me. And I went home and I took it. And it was kind of, I couldn't tell if it was positive or negative. And I took a picture and I sent it to Sabrina. And because we've ran like 5,000 of these things, we were like, yeah, if that was a rhododendron, we would say it's positive, So then I took a PCR, but the tests work. They work pretty well. They're just kind of funky, But that's all we got for viruses. And we got no control for viruses. You know, you're throwing out the plants. There's no antiviral compounds for a plant. You know, the antiviral compounds for people are really expensive. You know, that for hepatitis B, it's 80 pills at $1,000 a pill, right? Yeah, and you know, and everybody's pissed off about that, but a liver transplant's $350,000. So Cepheid and Bioscience is like, you're getting a deal, man, you know? So anyway, yeah, those free pills, they ain't cheap. And so your taxpayer dollars. And so typical symptoms of sucking it. So you get a feel for fungus, bacteria, virus. Let me do like 4 million insects in like 3 minutes, right? So when you identify plants, you always go alternate or opposite leaf, right? And that's easy, right? You know, opposites boxwood, alternates holly, even though they kind of look alike. So with insects, it's sucking or chewing, right? And sucking pests, they feed with a stylet. And that straw, they jam it in and they suck up plant cell sap or xylem or phloem fluid, and that leaves a spot. And the spot's called stipple, right? And so I use 4-line plant bug because the stipple is big, it's easy to see. And so you back up and you see the stipple from where the feeding behavior took place, right? Now, they all don't look like this. Most of them have smaller stylets and are smaller insects. And so you get a lot of stipple, it'll take the color out, you know? And so you see this, but the thing is, mites will do this too. Looks the same as mites. With insects that suck plant cell sap or suck fluid, they make a lot of waste, right? Plant cell sap is not highly nutritious, so you got to eat a lot. And so with these insects, if you understand what kind of waste they make, then you can name the insect group. So you see the stipple and you know it's a sucking insect. And with this particular tree, I turned over the leaf, and on the bottom there's an oily fecal mass, and that poo is pretty typical of insects in the group Tingidae, the family Tingidae, which are all the lace bugs. So that, you know, even if I didn't have the insect, the sign, the poo, told me it was a Tingid, you know. And then I go, what lace bug gets on it? This was a Zelkova, you know, and elm lace bug, right? So we got there that way. So again, you know, recognizing that sucking insect, and then maybe, you know, sometimes it's wax, sometimes it's wool, sometimes it's honeydew, sometimes it's a little, it's a little bit of fecal mass like this. But again, that helps, right? So you compare that to the chewers. The chewers chew everything in sight, or they chew in specialized ways, right? So we first start with a chewer like this, right? A caterpillar. And you look at, and the example's not a caterpillar, it's actually a wasp, a sawfly. And these things eat everything in sight. And everybody knows what, like gypsy moths will defoliate the woods. Or down here, you get orange striped oakworms, right? You know, they flare up every few years, and they just eat all the leaves. And so that's easy. It's easy to see these things because they're gregarious and they're all over the tree and that sort of thing. Now, we got insects that feed in specialized ways. And one of those damage is called skeletonization. They chew all the tissue between the veins. Now, generally speaking, in New Jersey, if you see skeletonized leaves, it's Japanese beetle, right? Now, there's other insects that skeletonize, but they're specific to their host, like oak sawfly or elm leaf beetle or something like that. You know, the flea beetles in eggplants and stuff. They don't really skeletonize, but they chew holes, you know, and that's a pretty distinct thing. You know, so you go from something like that to like notch makers. You know, if you see notches on the edges of leaves, that's what weevils do. You know, so you see, that's like, you know, 1/10 of an inch tall annual bluegrass full of putting green, and it's got a notch, right? So you see a notch, what weevil does this plant get? You know, could it be black pine weevil? Could it be white pine weevil on a pine tree, notching the leaders or something like that? So you know, this typical symptom comes from the feeding or breeding behavior, and that points toward an insect family, right? So again, you know, sucking versus chewing, and then how does it chew, and what's it doing, right? Some things make nests, tent caterpillars, webworms, right? Easy. You got bagworms, stuff like that. You know, everybody knows what bagworms look like. You know, they make nests, right? So, and then the last nest makers are things like emerald ash borer, the borers, right? So you're looking at feeding and breeding behavior. So. Probably Japanese beetles. If they're leaving the veins and it's skeletonized, probably Japanese beetles. They have a lot of holes. There's a couple of sawflies that do that too. You know, yeah, you got it's insecticides. Yeah, and you know the big problem is you're growing roses, and Japanese beetles love roses, but so roses got a lot of insect and disease problems, and so that means if you're growing them and you want them to thrive, you're in a fungicide and insecticide protocol. You know, and that's why I say there's downside to it. You know, as nice as the upside is, they take a lot more work, right? And so that's the problem. And that's one of the things that I do talks to like landscape architects and stuff, and I try to say don't use these plants in your plants because they get a lot of problems. And it's all the plants everybody wants. It's like crab apples and pears and cherries and, you know, rhododendrons. But like if it's got a fruit name connected to it, you got to spray it all the time, or it's going to be damaged somehow. So roses is in that category, you know. And so again, it's just because it takes a lot of effort. All right, I'm doing good. Are you guys okay? I know I went through that fast, but I always go through that fast because I see people's eyes glaze over in that part. So I figure faster the better. Yes, because they don't reproduce on their own. They don't eat. They don't grow. They don't do anything, right? Except tell somebody else to make a copy. Yeah, it's kind of a weird thing. So if they're not alive, how do we categorize them? You know, yeah, the viruses are an anomaly. We don't know much about them. And we really only started understanding them over the last like 30, 40 years. So it's kind of a weird thing. Yeah, it's kind of cool. I got a giant article that tries to answer that question. So I could send it, but I can't even get through it. Anyway, all right. So what about physical injury, right? You know, and physical, I mean temperature and moisture stress, right? And so when we don't have enough water, and remember the fuchsia, for whatever reason, the plant's not getting enough water. And so we define drought as an imbalance between water going in and water going out. And that could be because the water, or it could be because it's stuck in the soil, or it could be because the roots aren't working. The symptom is always going to be the same. And so you look at wilting first. Everybody gets wilting. If it wilts long enough, then the cells on the outside edge, the last ones in line. It's like if we all go through the line and get coffee, I'm not going to get it, right? You know, because I'm last in line because I'm here. And so, you know, they scorch, and you get a tip and edge scorch that's generally on every leaf in the entire canopy, right? If it's hot and dry, or hot and wet, or cold and wet, or cold and dry, damage is worse, right? You can't really separate them, but when you have extremes of both, it's worse. And so conifers do it too. You can see how they wilted, and then the tip and edge scorch, and subacute levels will knock leaves off prematurely. You know, like ash trees, they'll drop leaves. You know, if it's hot and dry, they'll drop leaves in August. I got a cherry in front of my house. It's a giant cherry, and we got a rainstorm. It rained 9 inches, you know, in late July, and the tree dropped all its leaves. You know, it was just a temporary period of anaerobic conditions, and it dropped. So subacute levels, you know, and you see a lot of pines turning yellow on the inside, dropping needles in like September because they were stressed in July, you know, and so that cause and effect. Now, more acute levels of stress, longer periods of time, and then the plants start to die the same as the leaves, from the outside in and the top down. So if you see it doing this, I'd be thinking about moisture or root zone problems, right? Something like that. Now, the other thing about this is if you have things like this, usually it's the cumulative effects, right? Heat and drought and poor site, improper plant select, it's like a parking lot, Why are you doing this? It's highly disturbed site. You plant this stuff. This was in 2010. It didn't rain from it didn't rain for almost 2 months, from like Memorial Day into July. And the first week of July had 400-degree days. Like July 6, 2010 is the state record at 106 degrees, right? And this is what happens, And look at the trees over here in a better location are fine. They're tolerating it better. So cumulative effects. So site matters a lot for this stuff. So that's heat and drought. What about chemicals? Because chemicals can look a lot like drought stress because of the ones that get taken up and moved to the outside edges. So we have chemicals that burn by contact, like diquat. We have chemicals that move in the plants, and then we have chemicals that are plant hormone mimics, right? So the thing about chemicals is there's usually an applicator, right? And so you see stuff that looks like this, right? Yeah, drop spreader disease, and right, and right, and so it's so ask yourself, is it along a road? What to spray? And so here's an issue where the plant was burnt by contact. So we use horticultural oils for like mites and stuff like that. And if you apply it on a sunny day now, like you're putting them out now in the dormant season, if you apply it on a sunny day, it dries, it's fine. If you apply it on a cloudy day and it doesn't dry, it'll burn. Or if you don't get the rate right, it'll burn or something like that. And so burns by contact, like the diquat. I remember a guy, I went to this landscape I have a friend, and he said to me, he said, "Remember when you got me fired?" And I was like, I couldn't remember, but because I was on the site with his boss in Princeton, and they had these trees. There was like a 20-foot green fence, and they went out there with a horticultural oil, and he sprayed them. It looked like Zorro. It was like this. You could see the spray burn. I'm like, you burnt them. And his boss had to replace like $30,000 worth of plants. So somebody had to get fired, and it was the trigger man, and so, but it was burnt by contact, but you could see the pattern, and so like with the deer repellent thing, it was all the inside of the trees that got sprayed, and so that burnt by contact, cause and effect. Now here's this slide again, imprellis, uptake, and move to the outside edges, right? So that's important there. A lot of product. So understanding how the materials that you're spraying on the plants interact with the plants, like I can look at something and say, yeah, that was probably chemical, or that definitely isn't. Unless something was sprayed that we don't know about, then it probably wasn't. And that happens too. Somebody asked me about corn gluten meal, as an herbicide, and I went to a site where the trees weren't looking that good, and they had been using corn gluten meal. An organic lawn care guy was putting corn gluten meal on the ground, and looking at the ground, and I'm like, there's no weeds, man. And I'm like, and I'm looking at the trees, and they look like this, and I'm like, what's going on here? And the person that was spraying was spiking the mix of corn gluten meal with a product that wasn't organic, with imprellis, this product. And we caught them because of the damage to the trees wasn't corn gluten meal damage, and it wasn't, it was all, anyway, be careful. Sometimes stuff's in the tank that you don't know. And so if you understand what product is being used, you can look at the symptom and maybe get a good idea. Here's a good example. Dicamba is a product that you might use for dandelions or plantain, broadleaf weeds. In Weed Be Gone, it might be like 2,4-D and dicamba and MCPP or triclopyr. There's a couple of common herbicides. I know that's dicamba because it always cups leaves up like that. And I also know it's dicamba because I did that to my own plants. I had a hose end and I was spraying the plantain in my backyard, And I went and sprayed the paver patio because there was some weeds popping up and it drifted onto my hydrangeas. And so got a good picture and it was a conversation piece all summer, you know. But again, you know, you understand the chemicals. So that's the thing, contact, uptake, growth regulator, right? You know, what are you spraying and what does it do to the plant? And that's how you get to there. Now, de-icing salts burned by contact, right? You can see the pattern here. De-icing salts are also taken up by plants and moved out to the edges. You know, like if we put out a bunch of salt now, right, when everything's getting ready to break and it rains and it goes into the landscape and everything takes it up, you'll see the damage, right? You know, on 81 in Pennsylvania, there's a 50-car pileup. You know, I drive that route up to Syracuse a bunch, giving talks. I'm glad we Zoomed this year. I wasn't on the road. But if you drive up 81 going towards Syracuse, you can see the contact burn all along the trees, all along the highway, you know, because they use so much salt up there. So again, you know, just another illustration of contact versus uptake. Now, nutrients is a whole different ballgame because imbalances and toxicities and deficiencies kind of look the same. It's going to be a color problem, a growth problem, or a burn problem. So the only way you can really tell with nutrition is get soil tests, right? Now, there's exceptions like this one. This is iron binds up in the soil in a high pH soil, and these acid-loving plants that will have iron deficiency problem. I mean, that's a no-brainer. But that nobody can go and look at a plant and go, "Oh, that's calcium." You know, unless you're the expert on that particular plant, you might know. But generally, you got to get a soil test, right? So that's that. Now, what about this one? Mechanical. Breaks, bruises, wind, rain, snow, the wrath of God, you know. And so this is Hurricane Irene. This is why you don't want to plant pears. Yeah, they just break all the time. And so it got squished. The Snowtober, you know, probably wasn't as bad down here. Remember in October, like 7 or 8 years ago, it snowed. My power was out for 8 days and knocked over everything. How about if you do construction underneath the drip line? You know, see the tree, the drip line is as tall as the tree is, so it's out here somewhere. If you do construction under here, you're going to damage the roots. You know, changing the grade, putting in a driveway, fiber optic cable, you're going to get a cause and effect, right? It might take 5 years for it to happen, but you're going to get a cause and effect. So be really careful about that. You know, if the contractor says when he's building your house, I'm going to save the trees, I would say just go ahead and cut them down because I'm going to be paying for getting removed 5 years later, you know? And so you want to be careful about that. This is in Fort Lee, New Jersey. I don't know how to explain this except for somebody don't know how to plant stuff. And so like this tree is this, and then it's planted with this, like what is going on here? And so planting problems are huge, right? And so, you know, what did you do? Why'd you do it? How did you do it? What happened? What was the weather like? You know, mechanical injuries and all that stuff, you know, vole damage, you know, porcupines chew on trees. You believe it or not, we got porcupines in New Jersey. It's crazy what happened. So again, you know, that all goes in that category. All right, now here we go. I need about 10 minutes to go to finish this up. So we're almost at noon time, and we're only on step 3. I've been talking about this all along though, so I go fast through this. This is the circumstantial evidence, right? What about the site condition, drainage, exposure, soil tests, and all that stuff? You know, other infrastructure. So guess why these died? Can you see the cause here? Right? Texas hates the wet. Infrastructure problem. How about this? These Leyland cypress are all burned up. You can't see this on the other side. If you turn this direction away from those trees, there's about a 60-acre soybean field of open, and the wind just comes along the field and hits the windbreak, and 1 year it burnt them all up, right? So there's a cause and effect. You know, we get samples from, I got a guy in Atlantic City sending me trees all the time. It's always the ocean side that's burnt, right? And that's what he wants me to say. It's the ocean side, salt coming off the ocean. You know, after Irene, 7 miles inland we were seeing salt damage. You know, I went to a site and so that sort of, like, I don't know, it froze. I don't know what to say. Like, whose idea was this? Right? And so, and then you go to stuff like my mom's house, where that's Pachysandra, and that's a swimming pool, and it's in the shade. And it's always so wet here, it favored Vallotella, the disease. We created conditions that favor the pathogen. Here's another one like that too. Look where the damage is. It's always wet here underneath these trees, and they all have pythium. And you come out here where it's dry and there's no disease, right? So site condition matters. How about that one? Look at the roots. That's planted in that came out of a neighborhood that used to be a farm field. You know, 60 years of moldboard plow, and you got a plow pan about 18 inches, and you put a tree in there, and the roots hit that impermeable layer, and the roots do this. You know, stuff like that. Or how about that one? Right? 20 years in the ground. The trees are two different heights on different sides of the road. And I went there and I kept going root. So they got the skid steer, popped it right out of the ground. 20 years, popped it right out of the ground. And we couldn't even get the forks in the other tree in the ground. And then they put it back and pulled it out, and it looked like it never came out. It's like, but that site was where all the construction trailers were, right? And it was so compacted that the trees couldn't establish, but they survived because they were getting watered with this intense irrigation system. I mean, the site was pristine. The trees were just half the size. And so how do you know it unless you look? So that's the idea. Got to dig stuff up. What about the weather? What about the weather, right? So here's what we're doing this week. 70 degrees and 22 degrees. And everything, all the magnolias, everything that broke bud's going to cook, right? How about rainy springs means more fungal leaf spot diseases, right? So if we get a rainy spring, if we get drought stress or winter damage, we get more cankers and borers. If it's a warm winter, we get more mites. You know, we have predictive models for lots of disease and insects that use weather data. I mean, I can tell you the exact day that we're going to have brown patch on our turf grass farm. Like, seriously, I got students in there. I'm like, Thursday, we're going to have brown patch. You know, and I'm older than their parents now. So they don't think I'm like the weird dude listening to the Allman Brothers in my office, right? And so they're like, yeah, right. And then Thursday they come back in going, how'd you know? You know, yeah, you should listen to me right from the beginning. And but you know, it's weather data. And so we're following the weather. What about management practices? Like, how much mulch do you need? Yeah, Sabrina and I got paid to go to this site. We drove into the neighborhood and we started laughing and drove out, you know. And so like we didn't have to get out of the car. I knew what the problem was. And like how deep did you plant it? Too deep? You got crummy, that crummy mulch, you got landscape fabric, you know, everything's wrong here, you know. So that's something. Like, what about this? Look at this Christmas tree farm. We couldn't figure out what was going on here, and we got a soil test, and it was potassium deficiency. Look at the same field after 2 years. Look at the trees, man. They doubled in size. You know, just because we fixed the new, look, they're all yellow, suggesting nutrients. It's all the trees suggesting a pattern, reinforcing abiotic. We got a soil test, we got more information, and then circumstances was fertilized, and we solved the problem, right? You know, spray the tree, kill the grass. Yep. And then, so what did you do? Why'd you do it? When'd you do it? How'd you do it? You know, we want to know all of this stuff about the site, you know, because sometimes it's really tiny little inconsequential piece of information. I remember talking to a lady about her landscape, and I'm talking, I can't listen. And then finally she talks about, we got a new irrigation system. And then she's watering every day for like 45 minutes. And like suddenly after half an hour conversation, I realized she just loved these plants to death. Like, you know, it just gets you killed. And so like, you know, listen for that stuff, and circumstance might be the key to the disease outcome, or might be the problem outright, right? So all of that stuff matters a lot, even though I did it in 5 minutes, right? And then last thing here, how do I identify the sign? This is how we wrap it up. So a lot of stuff you can see, right? We saw bagworms, we saw webworms, we saw cedar apple rust, right? Easy stuff. You'll start to get used to that, right? This is red thread in turf grass, and it makes these little sclerotia look like red antlers coming out the leaf tips, right? Nothing else looks like this. It's really easy. Once you see it, and it's really common in New Jersey. So there's a lot of stuff you can just see in your hand. People are going to walk in with some sort of gall off an oak tree. Easy ID, right? If you can't, you need to look a little bit closer. So you need a hand lens, right? Or you need, that's just a fancy hand lens, a $5,000 hand lens. And it gets you from 20 power to 60 power. And so you see it like this, and you put it under the dissecting scope, and it looks like this, right? So we know it's a fungus. We know it's spewing. And I'm just going to take this and put it under another microscope and see the spores, right? And so, you know, get a hand lens, and you can see mite eggs. You can tell the difference between insects. This is millimeters. This guy brought a vial of insects in and said, I have an insect resistance problem, insecticide resistance problem to this one, right? I'm not getting any control. I'm spraying. And we poured them out, and it was like 99% this one, you know? And all he had to do was look a little closer and realize that he was spraying for the wrong insect. It wasn't insecticide resistance. It was timing. That was the problem. But he paid us $100 to tell him that because he didn't look close enough, right? So, you know, I throw that kind of money thing at you because, you know, sometimes it's just dumb. Take a closer look. And so, you know, we have a microscope where we can get even closer. So we look at like Rhizosphaera like this, and we see that under the dissecting scope or the hand lens, and then we get under the compound scope and we see this. Right now we're naming the pathogen. Now sometimes we need to help it. So we put it in a moist chamber. So we see all these spots, and you go, is this spray burn or is this a disease? And 24 hours on the wet paper towel in the Tupperware, and you got fuzz, right? So now you know it's a fungal disease. And so then you ask yourself, what fungal disease is going to make a black spot on the boxwood? And then you got a good educated guess, you know, boxwood blight. I'm just going to go here and give you a little bit more confidence, right? And say that we see the pathogen, right? So again, just looking a little bit closer. Sometimes we got to remove the critter from the plant. So we're defining the problem as a corm issue. We cut a slice. We cut it into pizza pieces, we throw it in bleach. Because if it's a fungus, and we think it's a fungus because it's discolored, right? The fungus is growing between the cells. So the bleach sterilizes the outside and all the contaminants. And if we do it right, we get it growing on potato dextrose agar, and we get the same fungus. And then we go back to the microscope and try to identify it from there, right? If we do it wrong, and I don't want to say wrong, sometimes it doesn't come out the way we plan, right? And here's an example of bacterial culture. Like, look at all those different colors. All of them are different bacteria. So now you got to ask yourself, which one of them is the problem, right? So using these culture techniques are kind of funky. So we have to have targets. And with the targets, we'll pick medias that select for different pathogens or make them look different. Like, look at this one. You put under a black light and the pathogens turn fluorescent. Like, if I add V8 juice to the media, Phytophthora really likes to grow on it. It's a good way to separate Phytophthora. So, but we have to have a target, right? Now, this is fraught with problems, and it takes a long time sometimes. It could take weeks to get to an answer doing this. So we have antibody-based test kits for a lot of the really common stuff, a lot of bacterial and viruses, some fungi, and that's 15 minutes, right? You know, but again, only one pathogen. We have to have a target. And if we need real confidence that we're positive, we got to go to the PCR testing, right? And that's 100% confidence. So you're sort of thinking like this: 100% confidence, 95% confidence, 90% confidence. You get back to the microscope, you know, 85%. You get back to a fuzz on a leaf, 80% confidence. Ask yourself who you're working for. Do they need more confidence? If it's somebody's lawn, you know, where you're just going to fertilize more, cut a branch off of dogwood, 80% is probably good. But if one of your nursery guys over here, you know, I need to have a 95% confidence. And if it's a problem where, you know, I have problems sometimes that are coming out of boats at the port in Wilmington, Delaware, you know, that they're going to turn away a tanker full of onions, you know, I gotta have backup. I gotta have a PCR, and I got University of Delaware and the University of Maryland say it too. You know, so, you know, we do what we have to do based on the clientele's needs, right? We go as far as we have to go under that. You know, so the question I think I would need to answer here is, how do we pick the appropriate protocol, you know, once we get there, right? And that's the same way we get you to the best educated guess, right? We evaluate the host plant and all its symptoms. We try to define the problem, right? We look at all the predisposing factors, all the circumstantial evidence that might add to the disease triangle. And then we get as close as we can get to the causal agent, right, to the sign. And what we're trying to do is build a case for the disease triangle. And the more information we have, the better picture that we can present and the stronger educated guess we can make. And then you can decide, do we need to get this to a person like me that has more sophisticated tools to absolutely give us more confidence in the diagnosis, right? And so with that, like, we're all doing the same thing for like most of the trip. You know, I just fly in first class on the way home while you're in the back, you know. I just flew from Denver first class. Ticket cost $1,500, but the back, sitting in the back, was $1,370. So I thought, well, I'm just gonna pay another $100 to sit in the front, you know. And so, yeah, that gas price, don't fly anywhere right now. And so, so the last slide here is this. Fungi, I had to go on short notice to see my kid. There was a little problem I had to fix. And so flying on short notice is a problem too, you know, and with $5 a gallon diesel fuel. But anyway, so fungi don't read the book. So it doesn't always look like the picture, doesn't always happen at the right time. There's always some, you know, it's nature. And so try to qualify it as nature. And so be ready for something to happen that's not expected. So be careful, don't jump to conclusions, keep an open mind. So one of the things that we do with the turf grass is we have the exact same protocol for every sample. And we do the exact same thing with every sample, and that eliminates bias. Because I'll get biased with stuff, you know. I know in certain times there will be these problems, but I got to evaluate, you know, with an open mind. And I think that's an important thing to keep in mind. Start from scratch. Even though you know it's going to be boxwood blight because it's the boxwood, pretend you don't know and do the protocols anyway, right? And that means you'll find, because a lot of times stuff pops up you don't expect. It's kind of interesting, you know, how we get surprised. And so the more information you have, the better. So that means communication with the clients and pictures and all that stuff. You know, check the weather, use your resources, look around the room. There are your resources. Sabrina, my assistant, has worked for me for 24 years. And so, you know, you guys are all married or something, you know, it's like I'm married at work too, you know, and so it's an argument all the time. And so Sabrina's a bickerer. And so we argue over stuff, but together we do it better than we do separately because we both have different perspectives and different ideas. So when you're doing this, if you're all working together, you're going to get to the answer better, I think. And then last, learn to make a decision, right? Some of us, you know, at 60 still can't decide. And right, I remember going to the flower show, and there was a booth of Rutgers Master Gardeners answering questions for people, and I was down the road trying to sell soil tests, and people would walk up and ask them questions, and all the people in the booth would point at me and point down, like nobody wanted to commit, you know, and I was like, no, go back down there and ask them, you know. And so, like, what's the worst thing that can happen? I mean, think about this. What if you're wrong and somebody's plant dies, right? No big deal. It's only a plant, right? It's not like your grandmother. It's only a plant, right? And so, like, here's my perspective. Like, killing stuff is the reason we garden, right? And that's the fun part. Like, because if you kill something, you get to go shopping, you get to dig a hole, you get to do a bunch of cool stuff. So with that, like, you know, learn to embrace death and destruction. And thank you for listening. So. Thank you. Anybody have any questions? Yes.
Let's assume that we know problem, like, just talking about biotic diseases, I think I know the answer, but what's the best way to dispose of all the stuff that you cleaned away?
That's a great question.
Compost pile, obviously.
The answer is this. Do you have a pile or do you have a compost
I
pile?
have both.
Right? The pile, there's pathogens like some of these canker fungi that'll probably survive. Like boxwood blight makes a structure called a pseudosclerotia, will survive. But if you get that in the compost where it's cooking and it's practically on fire, then most of the stuff is going to perish that way. But so the short answer is this: if you're cooking, most of the herbaceous plant material can go in there and it'll be fine. The woody stuff I'd be careful about, and if you want to hedge toward not having any problems, send it to the landfill. Put it in a bag and send it to the landfill.
Okay. All right.
Yeah, that's the safest solution. Because you want in your vegetable gardens too, right? There's stuff like bacterial canker in tomatoes that's going to persist on your steaks, and if you turn stuff under, you're going to have problems that are ongoing. So if you're losing your tomatoes, I generally wouldn't put them in the compost. I'd put them in the trash. I'd bring my leaf clippings and stuff like that and use that in my garden. Yeah, powdery mildew is something that will survive pretty well on. Yeah, it probably won't compost well, but it'll survive on the ground on leaf litter and stuff. Yeah, but if you get it in the compost and turn it and it'll probably be all right. The other thing about something like powdery mildew too, it's a wind-blown pathogen. So no matter what you do, more is blowing in, right? So you might not be able to do anything about it. We always say leaf diseases that to rake the leaves, for trees and stuff. And I was doing a talk in Yankee Stadium, and I said that, and a guy stood up and started yelling at me, a landscape guy. And because in Westchester, New York, and Scarsdale, and those cities, you're not allowed to move plant biomass off each property. It has to stay on the property. And these old neighborhoods with giant shade trees got leaves like crazy. And so he wanted me to write a letter to the town council that said they had to rake the leaves or they were going to spray the trees with fungicides, right? And like, I can't write a letter like that unless I got peer review research to cite, right? So I started hunting to try to help this dude, and I couldn't find anything. So I started calling my colleagues, and I finally got to this guy named George Hudler, and Dr. Hudler's at Cornell, and he's the preeminent forest pathologist, at least he was. And George starts cracking up. He's like, Rich, he's like, there's no research. How are you going to control a pathogen that blows in the wind 50 miles? And he's like, that's just something we tell people to make them feel good. They think they're doing something. So sanitation is good, but there's a limit to its effectiveness, So yeah, how's that for an answer? Black spot on roses, that just, start spraying them as soon as they start breaking bud. And spray them, every week, every Monday. And I think the product that would be most effective is probably chlorothalonil, Daconil. Immunox probably has an insecticide put in it too. And yeah, chlorothalonil is a traditional synthetic fungicide. I mean, if you want something that's like organic approved and OMRI approved, the only thing that would work would be copper. I would think spraying copper every Monday, and that has its own problems. Right. Yeah, there's a lot of interest in that, and I actually have a talk about lawn care that talks about that in the beginning. And I went to Boulder, Colorado I was in Boulder last week with my kid, and they have a lot of alternative landscape where it's weeds, and I'm not saying weeds, but like intentionally planting weedy plants, we might call some of them perennials, and they select the weeds for weeds that are like Queen Anne's lace or something like, so they go in there and selectively pull out the stuff they don't want and let the other stuff take over. And it's really attractive. Now, in Princeton, at the university campus, they're using a lot of clovers. And so, yeah, it's pretty attractive. And there's a woman named Paige Boyle at University of Utah, that's studying the different kinds of clovers in turf grass mixes. Because in the 60s, we used to have clover in the turf grass mixes, right? So there's a lot of emphasis on alternatives, shrinking landscapes and that sort of stuff. Now, here's the downside. Turf grass is a really good nutrient sink and a real good carbon storage sink. Grass stores carbon better than woods, because when grass stores it, it stays, and woods die and release it, right? So lawns aren't as bad as people think, right? And so the question that we have to ask is, how can we maintain a lawn the way we like to see it, in the traditional historic sense over the last 50 years, without ruining all the benefits? The benefits are really well documented, and as far, there's a lot. I can do an hour on the benefits. And so what we're finding is, one is some of these alternatives, adding clover, shrinking, but like if you use tall fescue needs less water, needs less fertilizer. If you mow it at 3 inches and let it grow to 4.5 and mow it back down to 3, you only have to mow it every 12 to 15 days, right? So you use less fuel to mow it. And that's like with zero impact in visual quality. So my friends at Cornell have done a lot of work with this. Like an established tall fescue lawn only needs about 2 pounds of nitrogen a year. And in order to prevent runoff, the defensible amount of nitrogen on a lawn to prevent runoff is 2.75 pounds. So you're below that, so nothing runs off. Think about that drop spreader disease. None of that nitrogen moved, right? So if you put it in the system at the right amount, then it's not going to run off. So so there's a lot of environmentally consistent things that we can do to grow grass, and still get the same visual quality. I'm really interested in what's the least amount we can do and still get the same result. And my friend Frank Rossi at Cornell has been working on that his whole career. And like tall fescue, 2 pounds of nitrogen, mowing it higher, are all really, really good tools. So you can have lawns without feeling guilty about it, And so again, that doesn't preclude the idea of shrinking, and runoff from landscape beds is worse than runoff from turf grass areas. So sometimes making bigger landscape beds actually degrades the environmental benefits of your landscape rather than enhances them. And that's data too from Suffolk County, New York. It all goes back to my friends at Cornell that they're way progressive into this kind of like, they call it environmental services. And they actually have a specialist in New York City, working in all the cemeteries to try to, how can we enhance the environmental services of the parks and the green spaces to help overcome the paved impermeable surfaces of the city, right? So it's, there's people trying to figure this out. So
if you are not a lawn freak and you just want a healthy lawn, what kind of grass seed would you recommend?
I always recommend tall fescue, turf type tall fescues, because they have massive root systems and they need a lot less. If you fertilize them too much, they're not going to do well. So like last summer, it rained often enough, you would have never had to water tall fescue. It would have stayed green the whole summer, right? And like, so what we would recommend with an established tall fescue lawn is putting out nitrogen on Memorial Day and Labor Day, like a slow release form, 2 pounds, leave the clipping so they don't go to the landfills, mow it every 2 weeks, right? And like never water it. It's going to look really pretty good. For most of these like rural areas, larger land masses, let it go. let it go, you know, and it really works well. I got pictures where tall fescue's being fertilized at 0 pounds, 2 pounds, 3 pounds, 4 pounds. And you can't tell the difference between the plots from 0 and 4 pounds of nitrogen. So why are we using 4 pounds of nitrogen? And then like soil testing too. You send the soil test to Rutgers. They're going to tell you you need so much phosphorus and potassium. Well, there's a whole school of thought that they recommend based on what's called the sustainable levels of available nutrients. There's a whole school of thought called the minimum sustainable levels of nutrients, right? And these guys are trying to figure out what's the least amount. How much does the grass need? How much do we have to keep in reserve in the soil? And the sustainable level means you got like $500 in your wallet. And the minimum sustainable level means you got $20 in your wallet. And what they say is that turf grass doesn't really need all that nutrients. Like you never have to apply calcium. And you never have to apply phosphorus or potassium. You're going to get no growth response, right? So like they're telling you to use way more fertilizer than the grass needs. So I'm sitting here thinking that most landscape turf, getting a soil test is just a dumb idea because they're just going to tell you to use stuff. So I tell the landscape guys, because I can't like stand up in public like I'm doing now, you know, and say don't listen to them. I tell landscape guys, get a soil test so you find out what you don't need, right? And in our soil, if you look at our data, I got data in a talk from 2018. It's like 85% of the samples already have sustainable levels in the high $500 range. Because people have been putting like 10, 10, 10 on their lawn for 30 years, and there's so much phosphorus there. It's like, yeah, it'll be 100 years before you even have to think about putting fun. And like calcium, there's enough calcium in your irrigation water that you'll never have to fertilize with calcium. Never have to use calcium, right? Because grass hardly uses that at all. So you know you know, so this idea where we need fertilizers and like, you know, if you're growing tall fescue doesn't get very many diseases, doesn't get very many insects. You'll never have to use a fungicide or insecticide on a 3-inch tall fescue lawn. Never. I would buy nitrogen. That's it. And I would go to like the southern states or the Agway and buy agriculture grade urea. It's 4800. It's the cheapest possible nitrogen source. You know, get one that has a polymer coating or something. It might cost a little bit more, and it'll last longer. You know, it's slow release. You know, and that's all you need. And even the golf guys now, they buy the cheapest urea possible, and because the farm grade urea got rocks in it and stuff. You know, so they screen it and melt it in the tank and spray it out their sprayers. And they reduce their nitrogen, you know, costs by like thousands and thousands of dollars. You know, so pick the cheapest nitrogen and with nothing else in it. You know, if your pH goes below 5, sometimes that's a problem if you've got Kentucky bluegrass. So maybe every once in a while pH test is okay. You know, so that's probably the most valuable tool in your soil test for a lawn. You know, I'm not saying for tomatoes and stuff. Like I did 3 hours on how to grow grass for Ocean County, and everybody was just messed up. Yeah. Yeah. Yeah, I would not use perennial rye because if you use more than about 10% by weight perennial ryegrass, you're going to get 100% perennial ryegrass because it comes up so fast. So what with tall fescues, Kentucky bluegrass, about 10% Kentucky bluegrass, that because tall fescues are clumpy, right? And the bluegrass fills it in. And what happens over time is as you manage tall fescue, because Kentucky bluegrass needs more water and stuff, the bluegrass will fade away and the tall fescue will fill in. And so like if you go to Tuckahoe or something and get tall fescue sod, it's got about 10% bluegrass in it so they can cut it as sod. You know, that's a good way of starting. It makes a nice grass. It really is attractive. So, yeah. No, there's a bunch of good ones on the market. There's probably 70 good varieties. Yeah, Rebel is an old variety, but there's a bunch of them. There's a thing called the NTEC, the National Turf Grass Evaluation Program. If you go to their website, you can see rankings of tall fescues. Okay.
Oh, you have that? Sure it's not
I'd be
stale?
careful about fruit trees. If you want a small tree,
Probably they'll.
a big tree,
I missed the first 10 minutes. What did he say about?
yeah, I mean, I like some of the maples, like the red maples, like, like October glory maples. They have really good color. They grow well here. Something like that's gonna be a big shade tree. For a smaller tree,